Okay, thank you all for coming back today for the continuing of the statistic and say stochastic by extension session of this morning. And so we will start with Flavio Roche, who is coming, I'm not sure of the pronunciation, I'm sorry, who is coming from Sao Paulo, and he will speak about self-organized criticality for Galvez-Luchard-Bach models.
Thank you, Patricia. Can you hear me? Yeah. Well, good morning, everyone. And as the Patricia said, I am Flavio Roche, and I came from Sao Paulo. I work on the Neuromat, which is the research center of neuromathematics linked to University of Sao Paulo. In the next minute, I will talk about some of my preliminary results in my research theme, whose the title is Self-Organized Criticality in Hierarchical Modular Networks of Galvez-Luchard-Bach Neurons. And for such that, I divided my speech, my talk, into 4 parts. I will start with a brief overview and motivation, talking about some considering, and then I will present the network architecture and talk about the network itself, and the implementation of algorithm, the implementation algorithm, then the network dynamic, presenting the stochastic neurons, and at last, the results about the static model. So, some considering. Experimental study generally agree that the cortical networks have a hierarchical modular architecture. On the other hand, experimental study were able to measure neuronal avalanches, which is responsible for self-organized the system into a critical point. The neuronal avalanches obeying a power law distribution is a signature of self-organized criticality. And then after this considering, the following question arises. Can we construct hierarchical modular cortical network models that self-organize to a critical point with neuronal avalanches with power law distributions? That's our main question that we are interested to answer. So, about our network architecture. We started with a random network of n equals 2 elevated to 10 neurons. The neurons were connected with connectivity epsilon equals 0.01. And here epsilon gives the synaptic probability of any pairs of neurons in the network. This type of network is called a network of hierarchical level, or each equals 0. And the ratio in the network, the ratio of excitatory to inhibitory neurons is 4:1, which is based on the evidence that approximately 20% of cortical neurons are inhibitory. So the implementation algorithm of the network. Networks of higher hierarchical levels are generated by the following algorithm. First, we randomly divide each module of the network into 2 modules of equal size. And then with probability Ri or Ei, we replace each intermodular connection, Ij, by a new connection between I and K, where K is a randomly chosen neuron from the same modules as I. And recursively, we apply the step 1 and 2 to build networks of higher hierarchical levels, H equals 2, 3, and so on. And the network with a hierarchical level H has, hence, 2 elevated to H modules. And here in the top row, we might see a schematic representation of the several hierarchical modular networks. For H equals 0, the network is the Erdős-Henri type, and as long as the hierarchical levels of the network increase, the number of modules of neurons increases as well. These modules have a hierarchy of connections. Modules that are topologically closer are denser than between more topological distant ones. For instance, H equals 3, these 2 modules represent 2 closer modules than those 2 modules. And here in the bottom row, we have the respective adjacency matrix. This type of matrix shows which kind of synapses each neuron in the network realizes to the other neurons in the network. And hence, here, the blue dots represent the excitatory connections, and whereas the red dot represents the inhibitory ones. Oh, yes. And the information that the figure highlights is that the inhibitory connections can occur only inside of each module, while the excitatory connections can occur inside and between the module. And so here we can see the inhibitory and excitatory connections, and here we can see the excitatory connections between these two modules, and so on. So we used in our work RE equals 0.9 and Ri equals 1. So as I said, so the intermodular connections are exclusively excitatory. As we can see, as h increases, the number of modules increases as well, and modules are encapsulated in groups of modules. Inhibitory connections occur, as I said, strictly within modules, and so they are local, while excitatory connections can be both local and long range. About the network dynamics, the stochastic neurons. The Gauss-Leschbar neurons introduces stochasticity in their fire neurons in a different way. Instead of noise inputs, they assume that the firing of the neurons is a random event, whose the probability of occurrence is a firing function. Depends on the membrane potential, V. And so, by subsumming all sources of randomness into a single function, the GL neuron model simplifies analysis and simulations of noise spiking neuronal network. So, let xi be a firing indicator. If xi of t equals 1, neuron I spikes at the time t. Otherwise, does not spike. In that work, the firing function adopted depends on the membrane potential of the I neuron, Vi of t, and the neuronal gain of the neuron I at the time t, gamma I of t. For the mathematical convenience, we use the rational function given by the equation 1. And the membrane potential evolves according to the equation 2. And you can think, if the neuron J fires, then xj of t equals 1. This event increments by a wij the potential of every postsynaptic neuron that has not fired at the time t. The potential of the non-firing neuron may also integrate an external stimuli, I, and a part of these increments, the potential of the non-firing neuron decays towards 0 at each time step by a factor mu. Is an element of 0 and 1 towards to 0. So mu here models the effect of current leakage. So this model of the neurons is a LIF, leakage integrating fire, stochastic. So as I said, mu is an element of 0 and 1, is the leakage parameter, and I the external input. The synaptic weights are wij, positive for excitatory and negative for inhibitory synapses. And their magnitudes is W. We adopted in this work two homeostatic mechanisms in order to turning the critical point into an attractive fixed point of some homeostatic synapses, homeostatic dynamics, sorry. So the two mechanism is the dynamic synapses, given by the equation 3, and the neuronal gain, given by the equation 4. Note the neuronal gain enter in the equation 1 in the firing function. So the variable tau is your recovering time. A is the asymptotic value, the same for inhibitory and excitatory synapses. And U, larger than 0 and less than 1, is the fraction of synaptic weight lost when the presynaptic neuron J fires. I mean, when xj equals 1. The static model. We call static model the model in which mu is equal to 0 and there is no external stimuli. I mean, I equals 0. In such case, the firing rate, the activity of the neuron I, is given by the sum over all presynaptic neuron J, neighbor of the neuron I. Here, K is the number of neighboring neurons of the neuron I. And the activity of the network is given by the sum of the activities of all neurons in the network. It's the sum of all firing rates. Now the results. A previous mean field analysis of the model without inhibition has shown that the product gamma times W equals 1 is a critical line of a continuous absorbing state transition. And here we can see, independently of the hierarchical levels of the network, independently of modularity, the absorbing state gamma 0 equals 0 loses stability after the product is larger than the parameters, the critical parameters gamma critical, W critical equals 1. This is the first characteristic, suggestive criticality. On the other hand, the activity of the networks increase as long as we increase the modularity of the network. This occurs because since the connections are rearranged inside of each module, the neurons are rearranged inside of each module, the connections increase in each module, and so hence the activity increase. And here we calculate the avalanche size distributions for different levels, and we know that as long as the hierarchical levels of the network increase, we kind of lost the power law distributions behavior. So here for h equals 0, we have a value of the exponent of the power law distribution approximately 1.5 for h equals 0, which is suggestive of criticality. And when I say about the power law distributions and avalanche, in neuronal avalanche, I would like to present you a didactic example. So we have here 3 types or 3 sizes of the neuronal avalanches for the h equals 0. 3 types of avalanches with different size and duration. We can see the activity propagates in the cascade of events. Here's the one. Small. And bigger one. And so the exponent that represents this behavior is approximately 1.5 at last. The bigger. right? And so, as long as the modularity increases, the behavior of the system becomes subcritical, as we can see through the tails of the distributions. Begins to be a subcritical. And here, the duration, the avalanche duration distributions, and we can see almost the same thing for the h equals 0, tau d equals approximately -2 is suggestive of criticality. And when the modularity increases, the system becomes more and more subcritical, losing the power law distribution behavior. So here is exponential behavior. And at last, I would like to show to you the fine-tuning of model. Here we have a dynamical case, the temporal evolution of the neuronal gain. The dashed line represents the critical value for the gamma. And so we can see from this figure is that the system, the dynamic system, is not a critical or subcritical, but is a supercritical. So here we present a self-organized supercriticality because the gamma here is towards to gamma larger than 1. And I think that is it. I would like to formally thank to FAPESP, especially for the Neuromat, and for FAPESP for the grant, and my reference, and thank you for your attention.
Thank you, Flavio. Any questions or comments?
A question. The first bifurcation you showed, you had half a pitchwork. Could you please, yes, this one. Could you please explain again the role it plays in your analysis?
Yes. Sorry. Activity here is the order parameter of the system. The order parameter of the system. Which this figure highlight is when is occurrence the phase transition between two states. Between the absorbing state, there is no activity, activity is equal 0, and when we have the activity phase. As I said, works showing mean field analysis for every type of models without inhibition, without inhibitory connections, has shown that for gamma times W equals 1 is a critical line. So for W larger than 1, we expected we see a phase transition. And so here represent some type of criticality of the system. We don't know if the system is supercritical or subcritical or critical, but is an evidence of criticality.
Other questions?
Thank you for your talk. I found very interesting to study the modular architecture because for neurobiologists, something that I've been always surprised is concerning the pathophysiology. I want to say, so for example, in some pathology like Alzheimer's disease, where we have degeneration, is specific to cortex or to the entrance of the hippocampus, which means the entorhinal area. And we really think that the architecture is behind this fragility of the brain. For some also other pathology like epilepsy. So the way that the neurons are arranged, we will have
some kind of degenerescence.
Yes, will have an influence on the propagation of the epilepsy.
Yeah.
So I really think that is very interesting to study this, and it could have many consequences on the physiopathology. So thank you.
You're welcome. If I'm allowed to say, I show to you just the static model. You have to see that I don't present the dynamic results. And I think an interesting thing is that when we applied these two mechanisms in the architecture for different external stimuli, we can see a synchronization pattern inside of each module. For instance, for h equals 3, we have 8 modules of the neuron. And so each module synchronizes your activity in a different way. Is a quite interesting results there where so far I don't understand what's going on in the network for this. So it's pretty interesting. Thank you for your argument.
Yeah, I have a related question about how you generate the connectivity, in the sense that it appears to be very homogeneous pattern. And so do you think you have more not inhomogeneous, heterogeneous patterns? And is it related to something you think about?
It's a good question. Thank you for that. The network, there is no homogeneity. Is a network, is a sparsely random connected network.
Can you go back and show how you generate this connectivity?
Here or here? Here, I think. Is a random network, sparsely connected. And so I think that is not homogeneity.
Do you have a probability of connection?
Because we divide each module randomly.
Yeah. It's the same.
All right.
It is the same probability
then yes,
that you have.
yes. I think that you mentioned the.
I mean, even if it is sparse, I don't say it's complete graph, but it's you have a probability of choosing or not the connections. It's kind of Erdős-Renyi, I'm not sure.
Yeah. Yes, we have a probability, a connectivity 0.01. And
yeah. So yeah, homogeneous in this sense that there is the same probability of choosing. And
yes. Yes.
It's okay. Thank you.
Thank you a lot. We need to go to the next speaker, but thanks again. So now we are hearing Tien Quang Phi from Université Côte d'Azur, who will speak about a new method to simulate point processes.
I don't have something to. Okay.
Oh, okay. So good morning, everyone. So I'm really happy to be here today. So today I will present a new method to simulate point process. So my name is Pierre. This is work with Poincaré. He also here there today. So we both come from the Neuromode laboratory of University College. And I also work in the laboratory of the Neuroscience of University College. And so first, I will say something about point process. So for me, the point process is just a random collection of the points. So I will denote through the talk as xt for the collection of points before time t. Well, and then I will focus only on the point process having the conditional intensity. So that will that is defined through this relation. So you can imagine like it is a probability of having a new point in the small interval, giving the path before time t. And well. And more precisely, I will focus in this talk to the Hawk processes and have only self-exciting conditional intensity function. So why we interested in this model? So well, it fits well the model, the data, and also you see at each time you have a spike. The neuron Z emit a spike. It will modify the intensity function of the neuron I via the interaction function at Zi. And well, so somehow it will increase the chain to having a new spike of the neuron I. And well, for the simulation reason here, I will focus on the intensity bounded. So well, that means we will add a delay term, the red term here, to the linear Hawk process. So you can imagine that by adding this term, now you have you will have not no more than 2 points during a small interval during the small delay of delta. So that somehow gives you the idea to have the upper bound of the intensity. So now let's back to the simulation. Well, in the 1980, we have very popular method to simulate the point process. So it's called Ogata Algorithm, thinning algorithm. It says that if you have the intensity that is bounded by the positive number m here, so what we do is we just simulate the point, Poisson point process of the intensity m, and then at each point we will decide to accept or reject it with certain probability. And for example, you go to the point t1 and you decide to accept it with the intensity at time t1 and divide by the upper bound m, and go to the time t2. For example, I accept the t1, reject the t2, and accept the t3, and etc. Well, now we need one more ingredient for the new method, and here I would like to introduce the Kalikov decomposition. So while we focus, for example, we consider the linear Hawk process. And let's back one more time to this. So you see that at each time, you need to compute the intensity function. So now, for example, with linear Hawk process, you see that the red term, if you have a complete graph with a huge number of neurons. So this calculates this right term here will be very expensive. So that's why our solution here is we will introduce the random variable z here, and we say that we pick z equal to random neuron z with lambda z, and pick empty with lambda empty, and well, we can rewrite the intensity function as follow, and you see the first red term is that the constant, and the second red term is something is just depend on the activity of neuron Z. So well, that's why we denote the first term for phi of empty and the second term for phi of Z. And well, somehow you arrive to the rewrite the intensity at the expectation of something. And well, so the first remark is now instead of you taking all the information all over the neuron, so now you just need to pick a neuron at random. And the second thing to remark here is that, well, you see that you not only can decompose on the neuron, but also in the time interval. So that's why we introduce not very new, but the random neighborhood here. So for us, it's the couple of neuron and time interval. So for example, here, it might be one neuron with one interval. And the second might be two different neurons with two different intervals. Or maybe it will pick empty. It means that to decide to accept or reject this point, it does not depend on anything in the past or independent of the past. Well, so we pose a question that, well, in which condition we will obtain to this equation, or we can rewrite the intensity as the expectation of something. So to answer this, so well, trivially, you see that as soon as you can rewrite it as a convex decomposition, or we call it more precisely Kalikov decomposition. So here, lambda is the polarity distribution over the neighborhood family, and the phi v here I denote for the function that only depend on the neuron inside of the neighborhood v. And well, now for we're back to the new method, the simulation method, the new method here, and we call it Ogata algorithm. And well, we keep the first step. We simulate the Poisson point process on each neuron. For example, 3 neurons here. We simulate on it the Poisson process. And then the second step, the new step here, is to each point. Now you will pick a random neighborhood. And then you will decide to accept or reject it, the thinning step. And well, but you see that maybe if you you begin with this point in the first round, so maybe the thinning mark of the 2 points here in the neighborhood of each neighborhood, you don't know the each thinning mark, their thinning mark. So this quantity, this polarity become unknown. So how we solve this problem? So very naturally, you would like to sort it. So somehow you will begin with the point that is minimum in time, and just go until the last point. So you will be sure that you always calculate this quantity, this polarity, phi v over M, sorry for that, not gamma, but M, the upper bound of the intensity. Well, so the second method we propose here, and well, you don't know this quantity, you will replace it by interval somehow. Well, we will introduce the V-mean we will assume that we will reject all the unknown points in this interval V, and we introduce the V-max by accepting them. So well, so somehow you can introduce P-mean and P-max by V-mean and V-max, and then you have that P will be included in this interval P-mean and P-max that you know, P-mean and P-max. And well, so you also know that from thinning algorithm, Ogata algorithm, that you will accept a point if you draw a uniform random variable U here, this is less than P. So this means that even you don't know P, the precise value of P, but you still can decide to accept or reject it by this. If U is less than P mean, you will accept, and U greater than P max, you will decide to reject it. And well, to conclude that we have proposed 2 new algorithms for the whole process, and we even can run it in parallel. And actually, Paul had do it in parallel, and we can do this up to 100 of million neurons in complete graph. And so here is a graph of execution time of the algorithm. And so you see that the Ogata, maybe blue is very bad, even in million of neurons. It goes very fast to explode. So somehow, and compared to the KO for the Ogata calico, and 2 population, it means that we divide the network by 2 population. And so you see that the Ogata calico decomposition is very well, better than the classical Ogata algorithm. So we hope to extend our algorithm to the unbounded intensity. That means you don't have the upper bound in advance. And the second remark is we can apply our algorithm in more general point process as soon as you have the calico decomposition. And well, here are the supervisor. Patricia and Eva is my supervisor, and Alexandre, Patricia is supervisor of Paul. And we would like to thank Curie to support us to come here today. So thank you for the attention.
Thank you. Questions or comments?
Thank you. Can you explain a little more how you split the computation when, okay, you explain your algorithm allows you to use, yes, parallel computation. Can you tell us a little more about the organization?
I think Paul may be more better to answer this question.
You want to answer?
So what was the question exactly? It's very basic at the moment. It's just a a simple Python package that allows to do multiprocessing. And in fact, the set of neurons is divided as a partition of several populations, here 2 and 4. It's because we do computation on a laptop, so there is no point to go more than 4 populations. And so I don't know if I can say more. Did I answer the question or? Okay.
Other questions or comments? Yes.
Thank you. You said that you made your computation for aux processes on the complete graph.
Yes.
Can you make also a general graph, and can you do the same Kalikov decomposition for any general graph of interaction?
Yes, for the Kalikov decomposition, we can do it for not in every graph. In fact, yes. I think it depends on how you define the neighborhood family. Is that is this? So yes. So somehow you can do the, as soon as you can do the Kalikov decomposition, and the answer is you can do it for any type of graph. Yeah.
Thank you. You didn't say anything about the kernel you used, because I guess in the traditional Ogata method will work more or less well depending on how often you have to reject. And that will depend on the kernel you're using. So what kind of kernel have been using? Yeah, the H function.
The condition on the H function, is that?
I mean, not the absolute condition, but when you compare a traditional Ogata algorithm with the new one you're proposing, I mean, I guess the performance, the gain in performance should depend potentially a lot on the H function you've been using.
Yes,
no?
Yes, that's true. Yeah, yeah. In fact, I think the computation time here is somehow I believe it depends on the choice of lambda, the weight, how you do the decomposition, and also I think I'm not sure to test the other case, but I think it's performed very well in this case, with this particular type of interaction function. But I don't know for the other interaction function, but I'm sure about that the Kalikov decomposition will help us to improve the Ogata classical algorithm. That's what I'm sure. And in particular case with it, here we choose maybe the exponential decay of a function interaction. So somehow I think it performed very well in this case, in our case. Yes.
Okay, other questions or comments? So thank you. And we pass to the last speaker. Okay, so now this is the turn of Antonio Carlos Costa from INS Paris, who will speak about Maximally predictive ensemble dynamics from data.
Okay, so thanks for the opportunity to speak. This is going to be something completely different. I hope you bear with me for a bit. I'm not going to show any neuroscience models, but I hope you can still, take something home from my talk. So I'm going to tell you a little bit about some of the work that I've done towards the end of my PhD at the University of Amsterdam. So now I'm a postdoc here at INS. But today I'll tell you about this maximally predictive dynamics from data. So as you can see in these videos, nature presents us with complex, multi-scale, unpredictable patterns that defy our understanding. Yet in physics, we've shown a tremendous success at building coarse-grained predictive theories from the principled integration of macroscopic degrees of freedom. So one example of that is, for example, Brownian motion, where by approximating the collisions experienced by a larger particle through Gaussian white noise, we can predict its larger-scale diffusive properties. Okay. Now, in order to do this, we generally need either some understanding of the microscopic dynamics or some notion of symmetries or conservation laws that allows us to write down theory. Well, in most cases in data, we don't have this level of intuition. So what we'd like to do is to try to build still these coarse-grained predictive descriptions, but working directly from measurement data. And so in particular, I'm focused on systems from neuroscience, so either behavior or neural dynamics, where we've seen an increase in the amount of large-scale and high-resolution data, but these data are also very difficult to analyze. I mean, how do you make sense of things like that? Like one enticing hypothesis, one enticing idea that we're going to explore is to think about, let's say, behavior or coherent patterns in a neural network as evolving in some complex potential landscape where different wells correspond to different stereotype behaviors, and then fluctuations can drive the system to transition from one well to another. Now, in order to make this analogy precise, we need to be able to infer or extract some slow coarse-grained variables that capture the larger scale properties of the dynamics, such that then the faster dynamics can serve as fluctuations. And so what we're going to do is that we're going to draw inspiration from other fields, namely chemical kinetics, fluid flow, climate sciences, and we're going to use a transfer operator approach to search for such slow observables directly from data. And we're going to do that by studying the time evolution of ensembles or densities in a carefully defined state space. Now, the intuition for this is the following. So imagine you have water flowing down the river. Okay, so on a microscopic scale, the water molecules will just be bouncing around each other erratically on very fast time scales. But if you zoom out and look at a larger body of water, then you'll see that it's actually moving coherently down with the river. Okay, and so this is basically what we want to do. We want to, in some sense, integrate out or abstract from this fine scale randomness and see the larger scale structures, and we can do that by essentially zooming out and looking at the evolution of ensembles. Now, due to the conservation of probabilities, the evolution of densities is linear with time. So you can think about L, for example, as being the Fokker-Planck operator if you have stationary Gaussian random process. Okay, and so what this means is that we can, in principle, subsume the generally nonlinear dynamics of the underlying state space and their trajectories into the approximation of a single linear operator for the evolution of densities. Okay, so these two descriptions are equivalent to each other. But using this linear operator, then we can decompose the dynamics into a set of modes that relaxes to the steady state distribution of different timescales that are given by those eigenvalues there. And so the hope is that we can identify from these eigenvalues the fast decaying modes that represent, let's say, the fluctuations, and then the slow modes that capture the coarse-grained, longer time scale, larger scale properties of the system. Now, in general, when we're working from data, we're not guaranteed to measure all the variables that are relevant to explain the system, right? So we might be missing important degrees of freedom that give rise to complex, non-trivial, non-Markovian effects to the evolution of densities, or might preclude, let's say, metastable states that are living in a higher dimensional space that we don't know because we don't measure those variables, okay? So before we proceed, we kind of have to do some sort of, let's call it inverse physics, and try to learn from our incomplete measurements what is the full set of variables that characterizes the dynamics, which is Markovian and maximally predictive of the future. Okay, so let's look at a physical example for intuition. So imagine you have a particle that it's hopping in a double well potential. So it's going to stay most of its time near each of these wells, and then it's going to hop around. And the dynamics in this case, we call it underdamped, meaning that inertial effects are important, so the dynamics is second order in the position. which means that you need not only the position to predict the future, but you also need to know the velocity, right? So if I just give you the position at some time t, you're not going to be able to tell me whether the particle is going to move to the left, right, up, or down. But if I give you also the direction of the velocity, then you can predict the future. Okay, so these two variables, the position velocity or position momentum, is what we call the phase space variables, okay? And they are Markovian and maximally predictive of the future. Now, let's make an exercise. Let's imagine that we don't know about the physics, and we're just measuring the position of the particle. Okay, how can we know that there are these extra degrees of freedom that we're missing? Well, one simple way to get back this velocity information is to just include time delays of our position measurements, right? So given enough time delays, then we can approximate the velocity pretty well. Okay, so this is what we're going to do. We're going to expand our state in time. So we're going, let's say in this case, from a one-dimensional signal to a k-dimensional signal, where k is the number of delays that we take. And we're going to try to see, study the predictability of this reconstructed state as we expand in time, until basically we can find the number of time delays that gives us a maximally predictive state, so that we have a correspondence between this delay space and the original full state space. Okay. So it turns out that this transfer operator formalism I just described before, it offers a direct means to the estimation of entropy rates, which is a fundamental measure of unpredictability. Okay, so the entropy rate essentially measures the entropy of the future states given the current state. Okay, so in some sense it tells you how broad are the possibilities of the future depending on where you are right now. Okay, and in practice, when we approximate this transfer operator, what we do is that we make tiny partitions in the phase space, okay, and then we count how many transitions occur between these different partitions, and we build a transition matrix. And you can see here that from that transition matrix, we can estimate directly the entropy rate. So here, Pij is one of those matrix elements, a transition matrix. And pi of I is the corresponding stationary distribution that you can obtain just by taking its first eigenvector, okay? So you can see that you can compute this entropy rate depending on how many delays you take, so how much you expand your state, and depending on how many partitions you take, so how finely you discretize your state. And generally, what we observe is that as you include these time delays into your state, you essentially narrow down your prediction of the future. Okay, so you're using past information to narrow down your prediction of the future. And if the system is Markovian on some time scale, then you should be able to see that after some amount of delays, you don't do any better at predicting the future. So you don't need to bother more about the past. And that's kind of what you see here in this plot, where I'm showing you the entropy rate as a function of how finely I discretize the state space for different values of this number of delays, okay? And you can see here that clearly just measuring the position, it's going to be very unpredictable. So this is k equals 1. But as soon as I start adding time delays, then you see that these curves will collapse. And at a certain k, basically, I have reconstructed the state space. I get this maximally predictive state, which we hope maps to a more Markovian description of the system. Okay, so now that we have this reconstructed state, we can build our transfer operator and look at its spectrum. And as expected in this case, then the slowest eigenvalues will correspond to the slow hopping between the two potential wells. Okay, and in fact, we can predict the hopping rate directly by just looking at those eigenvalues. And I'm showing you here that as a function of temperature. You can think of that as noise intensity. And the corresponding eigenfunction, so the eigenfunction that corresponds to these eigenvalues, it identifies these two wells, these two metastable states. You can see that the sign structure splits along the energy barrier, and negative values are on the right side and positive values are on the left side. Okay, so you can use these eigenfunctions to identify these coarse-grained metastable states. And this has been done in many fields, and what I'm going to show you now is how this can be applied also to a system that it's more relevant for neuroscience. And in particular, we're going to look at the naturalistic behavior of the C. elegans nematode. So C. elegans has been a model organism in biology for a very long time. We know a lot about its connectome, its genes, and we're interested in trying to understand how behavior is modulated on different time scales and length scales by this underlying genetic and neural machinery. Okay, so in a nutshell, the worms move by propagating waves along their body, which moves them forward, backwards, or turning in seemingly random sequences, and it is by sort of chaining these sequences or these movements in time that the worms change the way in which they navigate and explore an environment. Okay, so here this data is taken from some collaborators of ours in the University of Toronto, Will Ryu, And this little red box there on the right is the microscope that is following the worms around on the plate so that we get very high resolution images of the worm. Okay, so already here you can see that behavior, even at the level of just a nematode with 300 neurons, is very multi-scale in nature, right? So you can think about behavior at the scale of the fine-scale muscular activation by the neuromuscular junctions, or you can think about behavior at the scale of like navigation, feeding, mating, or even longer things like aging, right? So it's very multi-scale in nature. And so in some ways it's analogous to trying to find these metastable states or rare transitions in a complex potential landscape. So let's see how this works for these worms. So first of all, we choose to study the behavior from a worm-centric point of view. So we remove the translational and rotational degrees of freedom, meaning we don't really care if the worm is moving forward to the left or forward to the right. We still want to call that moving forward, okay? And so basically, we parameterize the shape of the worm. And it turns out that at each frame, this shape of the worm is pretty well characterized by a linear combination of 5 primitive shapes. So basically, by changing these 5 numbers here, I can generate any type of shape that the worm can do. And so this means that I can translate a video like this into a time series like that, okay? A 5-dimensional time series that now I can use to do my analysis like I've done before. And so then we do the same that we've done for this double well potential. We add time delays to these measurements of the worm. And we see that after roughly about 0.5 seconds that these curves essentially collapse and that we have reconstructed the state space, okay? So the state of the worm now is composed of short posture sequences, short movements, short little waves, okay? And now given this transfer operator, we can do our spectral decomposition, and we actually find slow dynamics that evolve over time scales of order 10 seconds, so almost 2 orders of magnitude longer than the fine scale dynamics. And interestingly, these correspond to long time scale behavioral states that have been identified that C. elegans uses for navigation. Okay, so here I'm showing you a projection of the corresponding slow eigenfunction onto the space spanned by the body wave phase velocity, which captures the frequency of the worm moving forwards or backwards, and the curvature of the body. Okay, and you can see that on the one hand, this red patch here, negative values, correspond to the worm basically just moving forward with very little curvature. And in blue, so these positive values correspond to the worms moving backwards and then turning. And this is a maneuver that it's been coined a pirouette. And these 2 states are what the worm uses for navigation. So here you can see that clearly at the trajectory level. So this is the trajectory of the centroid of the worm, so where it's going. And you can see that in red, basically you have these runs that are interspersed by these pirouette events. So it's very similar to the kind of run and tumble description that we get for E. coli. But here it's for a multicellular organism with a brain. And remember also that I'm not using any information about its position, okay? I'm only using the posture itself to identify these states. So it's kind of surprising that you can get back these larger scale things. So if we now subdivide the dynamics into these 2 coarse-grained states, we find essentially 2 time scales, a longer one that extends to hundreds of seconds that corresponds to these long runs, and a shorter one that corresponds to these pirouettes, okay? So we get a way to do this principal coarse graining of behavior at this scale. But it turns out that the transfer operator is also a very simple but very accurate model of the dynamics, okay? So I can go now, take my transfer operator and just sample states according to the rules given by my transition matrix, okay? So essentially, I'm going to be diffusing in some high-dimensional state space, and we can map that to the postures. And it turns out that the simulations are very accurate at the level of the microscopic posture dynamics. So here I'm showing you the body curvature as a function of time for data and simulations, and on the right you have the corresponding kind of animated worms. You can already kind of see that the dynamics resembles between the simulation and the data. And on the other hand, besides capturing these finer scale dynamics, we can also capture longer time scale dynamics. So we can now put these worms on a plate and we can make them move, okay? And so what I'm showing you here is the position of the worm in the lab frame. So this is on a much longer time scale. We're talking about minutes now. So red and blue are, again, these 2 different states. And so we can generate these trajectories. And it turns out that these trajectories actually resemble really well what we observe from data. In fact, we can measure the mean square displacement for these trajectories and compare it between data and simulations. And we see that our simulations capture the full transition between ballistic motion and diffusive motion at larger time scales. So gray is the simulations, and the dashed black line is the data. And so we can even infer a diffusion constant, an effective diffusion constant in this diffusive regime. And we see that our model and the data match pretty well. So here just green is just a shuffle where we destroy the dynamics but keep the relative abundance of runs and pirouettes, and that makes a huge offset to the diffusion constant. But if we keep the full model, then we can predict this diffusion constant. And again, this is a very non-trivial jump in scales, okay, that it's obtained from a very simple model. It's just a Markov chain, but built from this reconstructed state space. So by thinking principally about the representation, we can simplify our theory and still be able to make pretty dramatic predictions and bridging timescales in behavior. So we're now interested in trying to understand, what kind of neural and genetic perturbations we can do to modify the behavior at these scales. Because we can study behavior, as I showed you, from the microscopic fine-scale postural movements all the way up to the way it navigates and explores the environment. And I'm also interested if you have any ideas of, examples from neuroscience or data where this kind of approach would be useful. I'd be happy to hear your thoughts on that. And I'll just leave you with our recent archive preprint, and I'd like to thank my collaborators and thank you for listening. Thank you.
Thanks a lot. Comments or questions?
Thanks for a great talk. I would like to ask you, how well does your transfer operator represent more than one C. elegans, let's say, and also whether there is a way in which you can incorporate sudden changes in macroscopic behavior. Let's say you have an animal that at some point decides to do something non-trivial, and so what happens to your transfer operator then?
So like this whole transfer operator description kind of assumes, in some ways, from the way that I'm doing it, that I'm in some kind of ergodicity limit, okay? That I've observed the dynamics for a long enough time such that I can capture its longer time scale properties. So whatever I'm inferring, it depends on the time scale of my measurements, right? So if I'm looking at a much longer time scale measurements where the worms actually do more non-trivial things on longer time scales, then I can pick up on those. But in this data set, it's just a foraging data set where the worms stay on the plate for half an hour, and these are the longer time scale things that happen. In fact, there is a slow transient to the dynamics that comes from the habituation on the plate that we cannot pick up from the transfer operator, okay? Because it's so slow. It's slow, like much slower than the measurement timescale, that I cannot really pick up on it. So essentially, you will find whatever that is in your data set. So yeah, if you have those types of things happening in your data set and you can sample them long enough, then you will be able to see them. Otherwise, no. Yeah. Another thing you can do is that you can sample across multiple worms, multiple experiments, and you can think about each worm as a different initial condition for your dynamical system, in which case then you will be able to see these longer time scale properties. Yeah, good question.
Yeah.
Thanks, that was really interesting. I was wondering how you're choosing the partition of the state space to come up with the transfer operator.
Okay, so that's an interesting point. So the partition, I do this maximally, I try to maximize the entropy of the partition. So given my data set, I devise some Voronoi cells, okay? And I try to make them fine enough to maximize the entropy, but not too fine so that I start having finite size effects. Okay, so there is a trade-off between how much data you have and how finely you can discretize your state. But what I try to do is to capture as much information as I can in my partition before I do this subdivisioning again. Yeah.
Thanks for the nice talk. I was just wondering, could you see collective behavior of your worms in this model?
Yeah, so that's a good point. We haven't tried to do that right now, but you could think about applying the same type of formalism to collectives of animals. And there is now data on this in worms, and actually worms do crazy collective behaviors. But we haven't done that yet. And to be honest, I'd have to think about how this would generalize to a collective. It's non-trivial due to the properties of how the densities will evolve. But it's a really good, interesting possibility that we'd like to explore. Yeah.
Okay, other question? Oh.
We did do this in collectives of neurons, though, which is kind of similar in the worm. And you also find the same types of long time scale neural states, so collective neural dynamics on long time scales. Yeah.
Thank you very much for your talk. I will have a very naive question and try also to connect to neurobiology. I was wondering, do you know, in terms of ethology, why these worms are doing this oscillation? Is it protective? Is it because it's the best way to track on your way any source of feeding, or
that's an interesting question. So we haven't actually analyzed whether, I think what you're asking is whether these kind of macroscopic states are optimal for like dispersal and foraging, or to trying to find food sources, or
because for me I always try to do connection, but so it really looks like an oscillation, and I was thinking, okay, maybe we could use this type of oscillation to reproduce that we have in the brain. And I was thinking,
I see.
Imagine that you have something like, I don't know, which arrive in your petri dish, like a wave of water or anything else. Could we simulate, I don't know, like something that happened in the brain and could affect this oscillation?
Yeah, you can.
I was thinking about a model like for disease in brain, like when you have an AVC, a stroke, and so you have blood arriving, just what's happening on the oscillation of the brain.
Yes. So that's a really good question. I mean, there's been some work on this, on more mechanistic models of the C. elegans brain. So the C. elegans neurons are a bit weird in the sense that most of them don't spike. They have a more kind of continuous activity. But some of them have been kind of identified as central pattern generators, so they keep some sort of frequency. And they keep a coupling between the head and the tail. And people have looked at different types of perturbations they can do on different neurons and how that impacts the behavior. But so, I mean, we are now starting to look at different types of perturbations, both genetic and neural perturbations, so either ablation experiments or mutation experiments. And we're seeing like very interesting changes in the behavior that come from, let's say, if I ablate a specific type of neuromodulator receptor, that's going to impact the behavior on a very long time scale. So we are starting to ask more kind of mechanistic questions about the underlying control mechanisms. Yes, that's a really good question.
Yeah, thank you for the talk. I just have a technical question about how you expand the state with delays, how you choose this. I was not sure.
So how do I expand the state with delays? So I basically just concatenate these delays in time. Okay, so I don't know if I have a. So let's say you have a time series that goes from x1 to xt. Okay, then for example, for k equals 2, I build a matrix like this. Okay, to xt minus 1. Just from a practical perspective, this is what you do. You expand the state, but you add a dimension that it's a temporal dimension. And actually, I mean, this type of approach has been, proved by mathematicians a long time ago to actually give you a precise mapping between this delay embedded space and the underlying phase space or the state space of the dynamical system. So that's the so-called Takens delay embedding theorem. Takens wrote it for deterministic systems that it's been expanded now to recently in the 2000s by Stark to general classes of stochastic dynamics. So you can basically build a mapping between the original, let's say, n-dimensional phase space and the delay embedded phase space such that 2 points are like diffeomorphic to each other, meaning like they are the same, and their neighborhoods are the same. So if you study the dynamics on one space or the other, you should be able to find the same properties. They preserve all the ergodic properties of the system. Yeah.
Thank you.
Okay, thank you. So right now we are doing a kind of experiment, which is the conclusion of the afternoon of yesterday and the talks of this morning around statistics and data and how they are useful for neuroscience. So we'd like to open the discussion maybe with Ingrid and Alex. and see how you would react and to the questions and how we can maybe build a good discussion around that. You want to come?
So
I think I should take this out.
you prefer to use
Here's the microphone.
So would you like to... We were wondering, do you want to discuss just about the few, I mean, the presentation of this morning, or are we trying to do a more general discussion about that we have seen yesterday and today? So that's why.
Okay, yeah, so as far as I can see, maybe there is a gap between some presentations, in the sense that some are accounting for learning, some are not accounting for learning, but in a sense there is a common approach trying to find patterns, okay, between structures and dynamics. And as we saw this morning, there is this notion of meta states, or searching for criticality in large neural networks, and at the same time trying to find metastates in the behaviors. And I mean, even yesterday we present something in the sense that finding these different patterns at the behavioral level during the learning process can help explaining the neural activity. And so, I don't know, maybe if you have any ideas of these links, I can see that there is maybe questions that are the same about populations this morning. There were populations in the simulation approach. There were populations or modules, it's called modules, in the self-organized criticality approach. And finally, we can see that there is this notion of bottom-up approach in the sense of finding meta-states, stable states, and so on. And so, who would be interested, I don't know, by the learning aspects of that? Or do you have any experience about that?
Okay, maybe I will change and be provocative in a different way. We are lucky to have biologists in the room, and we are also a bunch of mathematicians, statisticians, and stochastics. And yesterday, Cass told us that typically statisticians are very skeptical and don't trust models, whereas neuroscientists are boldly adventurous.
Can I add something?
Yeah.
I've been impressed, because as I said yesterday, I think I spent a very good time, and I really enjoy, and I really see the interdisciplinarity now. So it does mean that you know so well about all the work, all the brain works. So I've been really impressed, and so in the same way, I was wondering, what do you expect from the biologist? What do you need? What do you think that to contribute in the best way to progress in terms of knowledge? What would be interesting for you, and what are the next steps? Because I see that so far we have many biologists converted in mathematicians, and the other way also. So I think we are almost close to understand the functioning.
Nobody wants to talk or give their point of view? Yeah. Okay.
The obvious, from the mathematician modeler's side, the obvious thing we need is we come up with models, and these models make predictions. And obviously, we'd like you to help us check whether these predictions are correct, set up the experiments, the corresponding experiments, in a loop, in a closed loop discussion.
Definitely. I think is the way that we are trying to work together also we are making prediction together, and we see if, okay, in the physiology, does it work also in the same way? But something that I would like to point also is, today I realized that I think with the pathology we can also see the evolution of these dynamics, because it's true that all the talk were concentrated on the dynamics of the network. And so we have fabulous tools now in biology where we can change the activity and also change very specifically the activity with tools like optogenetic and things like that. We can inhibit some specific networks. So, for example, I was thinking about just the inhibitory cell or excitatory cell and see the repercussion. I was thinking about the colon and also your model. So we have these tools to just play with the models and see, okay, how does it work? And yeah, definitely we can do many things and see how the things evolve. Yeah.
So does it mean that one way to convince biologists that a model is good is that we make a prediction that you can verify? Or would it be the other way around, like we make a model which looks like what you are seeing, and then if it looks like, and what do you mean by looking like, it will convince you?
I think we have different approach in biology. So, for example, in my case, I think the simulation and the models are very useful to extrapolate to the entire brain, because unfortunately, when I'm studying, when I was looking at showing the tetrode specifically in the region of the brain, it was very local. And to extrapolate to the entire functioning, I don't see another way except the modeling and the prediction that you can do and that we can't. As you said, the second step, I think, is a good way to see, okay, you give some prediction, so now we're going back to the biology and see how does it work, does it fit to the model, and if we constrain the model, what's happening, and could predict some pathology or dysfunctioning and things like that. I think it's a good way also. As I said this morning with some of you, so the main problem that we still have is, I think the biologists are very it's not very good. It's very difficult to speak between disciplinary and to interact. And so the biologists are convinced. So, for example, yesterday when we have the talk of Cass, so I was really impressed, because in biology, all the people told me, okay, so if you're moving your electrode,
that's Eden.
It was Eden, sorry.
Yeah.
If you're moving your electrode, so you can't be sure that you are recording the same neuron, and in this case, you can't say anything about the learning phase, the evolution on the same neuron. And with the tools that that a mathematician propose, actually, I think we are closer and closer to see, okay, they belong to the same cell assembly, they have the same shape, they have the same pattern firing, they are firing on the same place, so we can admit that it probably belong to the same cell assembly and work together, and maybe it's the evolution of the neuron, and we can compare them. So, as I said, so we have data, and I think we are exploring our data even not at the half of that we got. So it's probably really something few, and is thanks to you that we can explore and go deeper on the analysis.
Because it was about statistics, maybe I would like to interview Christophe, who is a biologist who is now doing statistics and is also very fond of reproducibility. So maybe you have something to say about how statistics can help neuroscience and vice versa, somehow?
Yes, or at least the maths. I mean, I think we should be a bit more precise when we speak about maths from the biological side, because for biologists, as soon as you write down equations, you're doing maths. When you start talking to mathematicians, the first thing you have to learn is that to make a career in maths, you have to produce theorems. To produce theorems, you have to work on simple enough models. And that immediately creates, the first barrier between your experimental reality, your daily life as a biologist, and what the mathematician can do. By that, I mean, you have to set the question at the right level. And it's hard for us because right now, I guess, for us experimentalists, or for me, former experimentalists, we can observe things at too detailed and too sparse level to feed properly, to compare properly with what mathematicians could do. And that's, I'm not sure I'm clear. When we record several neurons at once, we always have a very sparse sampling of the whole network. And that makes, at the time when we want to compare the experimental data with what modelers or mathematicians can predict, there's a non-trivial choice to make and to decide what is comparable in the data with what the mathematicians have worked with. And so for me, it has been the largest, the main problem talking to mathematicians. And then to come back to your question, Patricia, about what's the use of statistics in neuroscience? The data are variable. Whatever you do, you never get twice the same thing. So when you don't have a choice. You have to look at population of experiments and characterize this population. So one way or another, you have to use statistics. Then the problems, so to speak, I would point to between statisticians and biologists is that the knowledge of statistics in biology lags a lot with respect to what you statisticians are doing. So the stats that students are learning now, and that's things that were done 50 years ago. And that doesn't mean that they should not learn that. It's still useful. But there's a whole lot of developments in the last 20, 30 years, mainly through non-parametric models, clustering techniques, and now machine learning, that it would be very useful, I think, to think on what kind of teaching should be brought to master students in biology, because they see that as something marvelous. They heard the keywords, they've seen papers where it's used, but they have no clue of how it's implemented, what it's worth. So there's this, and essentially these are two cultural gaps. The fact that when you talk to theorem-proving mathematicians, you need to present the problem so that they can prove theorems. And when you talk to more applied mathematicians working in statistics, they have very sophisticated tools that are potentially super useful in biology at large, but in neuroscience in particular. But people are not aware of that, biologists. So I don't have a solution, but that's the two problems I would think we should work on as mathematicians or statisticians is how we expose or present these techniques to biologists.
That is very good point, Christophe, because so for the biologist, my student, they just want the p-value, okay? And so they try all the statistic tests to have p-value, and so and that is significant. And the funny thing is, many times, because it's not fitting to the Gaussian, but just remove the outlier. But as I told them, as soon as you're doing in vivo, that means where you have behavior, we have different phenotype, we have different behavior even in humans. So we don't fit all on the Gaussian curve. And so we have to consider also the one outlier. As soon as I told them that if you use properly the statistics, you can include all your animals, or you can just, in the way that you ask the question, you can have an answer and it's become significant and you can have a p-value. At this point, it's coming interesting for them. But so we have to convince them that is a power. The statistic is not here to tell them, okay, you have the result that you were expecting, related to your hypothesis or not, and just change the mentality in the way like we are doing with Patricia. I was asking a question and say, are you able to ask that in statistics? Yes, we are. We have a test and we can just ask this question in this way. But it's true that we have to change the mentality, and the problem is also, I think we need more people like us who are doing both, just to train the student also in the right way. But
No,
I have a question from biologist to modeler. I was wondering, because all the time, people thinking, when I discuss with biology, they say, okay, you give the data to the statistician or mathematician, and so they treat your data. And actually, it's not true, but I was wondering, in which way can we help you to develop, to go deeper, to go further for your own model? It's also my question, because I'm always coming with my data and say, can you help me to interpret or to treat this data? But so in the other way, how can we help you also to have your own model or develop your algorithm and things like that?
Well, so maybe this arrives with another loop I was thinking of, because the theme was also machine learning, and we have seen this morning two talks about simulation. And right now we have the power, thanks to the computer, to simulate thousands, billions of models of neurons. And I'm not sure we know what to do with that. I mean, at one point, we need the biological input to tell us if the fact that we are able to do those simulations is something that is meaningful for you, or if this is something which is just a nice game that simulators are doing, and this has no impact on the way you would see now your whole system. You see what I mean? I'm a bit caricatural, but...
No, but I think so that I've seen from yesterday and today, so I think you are in the right way. So we are not interested in terms of increasing the power. It's not that, because the brain is not that. It's well organized. I don't remember, I think it was who said that yesterday. So it's not homogeneous, it's heterogeneous, and the activity is not the same in the hippocampus we have seen in the cortex and things like that. So increase the power is not enough. It's not that we want. That's why we have to combine architecture with a balance between excitatory and inhibitory to increase all this detail. Yesterday we saw about the CSD and all this idea to build the best model. And I don't think we I don't think the best model would be a model where you can implement everything inside. Is, as you said, it depends on the way that you ask the question. And so it's always like that. And I think it's the way that we ask the question that we can very precisely define a model.
Okay, thank you. Yes, Daniele. Alex a disparu.
Yeah, so in fact, my view is you asked earlier on, you know, what could be done at the interface, let's say, and my experience is to really try to sit down and come up with a research question. I think this is something that you've seen yesterday happening between the three of you that seemed to have picked up at some point because you came up with a research question, established scales that you are interested in, and that very often also points to a direction for the model to take. For instance, not all models are the same. Yesterday we saw spanning already mechanistic models or more data-informed type models, which are not necessarily written down as ODEs. This morning you have seen an example of a mix of both of them. And my viewpoint is very much in the way this can be done from biology to, from experimentalist to theory. It can be done the other way around, meaning that typically if I sit down with somebody that has data available, I have tools that are in need of data, as you were pointing out. And the discussion can happen in very much the same way, sitting down, formulating research questions, this time perhaps from the other way around, and asking for common protocols to just advance. And so your question was, in some sense, what can be done? And my point is, well, probably one thing to do is just sit down more and have more exchanges of this type.
You're right. But so when we start with Patricia, Alex, for example, so it was at the beginning the main problem that we had. It was a common language. So we need to learn first how to communicate together, and as soon as we got this, so it's the key for the rest of the thing. Yeah.
Okay. I don't know if someone wants to. Yeah, you want to add something.
That's a question. We talked about making measurements. What about stimulations? Putting things in a closed loop where you would both stimulate and measure and try and understand the system better. We know this exists, for example, for some kind of Parkinson diseases. Is this something you feel is important in the future?
Definitely. It's going in the same way for me than I was explaining about modify the system to see if the model can apprehend and evolve with that. So, for example, in our collaboration, so we want to. Okay. So we're going to implant an electrode in the hippocampus and the prefrontal cortex. So now you know everyone where is it, and so we have a specific monosynaptic projection. So and we want to see if what's happening if I inactivate specifically with this one. So we have the possibility to stimulate closed loop and inhibitory loop, but so we have also the possibility, as I said just before, to inhibit specific projection. So it is in the same way, and I think is that which is very interesting, and I'm sure that you will convince the biologists, by the way, that your model is fitting to the physiology. Okay, that's fine, but as soon as you modify your physiology, is going in the same way for the model, and it's also the same prediction that we did with the cognitive behavior. So the animals behave really like that Alex was predicting with the model. So yes, stimulation is also one possibility, and change and modify. Yeah.
Yes.
Concerning the closed loop, I'll play the devil's advocate. So it's definitely interesting, but the danger is that when you're doing closed loop experiment. You're focusing on data you can make sense of. And my experience from the olfactory system is that for years we had, so to speak, old-fashioned experimental techniques, putting one electrode in a single neuron in an unstable condition. So we could sample very few neurons for very little time. And so we built from that a picture of how the first olfactory really works. The day we could put lots of electrodes and sample many neurons for a long time, some that did not seem to respond very strongly, but since we could simulate 50 times, 100 times, we realized that the system behaved in a much more subtle way than what we thought before. So it's very important, I think, to also keep a more exploratory approach that's not leading us to select data before we store it on the hard drive. And biologists are very keen and very good at looking at, finding patterns in data, and then they ignore all the other stuff they can't make sense of. And so we should keep also an approach where everything is stored, even if we don't understand.
Thank you, Christophe. Yes. Oh, but that's good. The discussion starts to take.
Actually, it's a very nice comment in the sense that, to my experience, it's true that I can see two different approaches. We were discussing about that in a lab meeting. Maybe you can have a naive approach where you try to find some patterns, taking some experiments, and you have generic tools, you apply them, and maybe something will come, a little bit as in our collaboration. We didn't expect that, so and so there is an emerging behavior. You find a new pattern, and so you can build a new hypothesis with biologists and so on. So which is okay, definitely maybe I don't know if it is better or not, but and the other approach. It is true that maybe there is it is dangerous, but in a sense we are discussing about, okay, maybe now we have a better understanding of some behavior, and we would like to build some new experiments in a closed loop to have a thinner explanation of these behaviors. And so maybe they are not opposed, and maybe they are complementary and different approaches. And it is true that sometimes when we presented some generic tools to people that are in, I would say, biostatistics, and they said, okay, but it's a general methodology you are doing. It's which part of the brain, which behavior do you want to show, and so on. And so, I mean, I can see from my experience that both approaches are interesting and complementary, and but definitely different people are applying these approaches.
Thank you.
What is your advice concerning that you said?
I don't know. If I knew, no, I don't know. I mean, it's important to have several different approaches and people with different bias. And we all have a bias when we tackle a problem, and it's good like that. I mean, as long as we keep talking and try to understand at the end what goes on, that's the way to progress. So I don't have any rule written in marble in the rock how we should proceed.
Because I think these approaches are complementary.
Yeah.
In the way that, okay, some, as you said, focus and don't see the forest behind them, but and the other one trying to integrate all the things. So yes, I think is for science, for me, is complementary, but I don't know.
I fully agree. That's one of the.
It's true that we all have a bias in our experiment, for sure.
But one of the key questions for me right now concerning my kind of specialty, which is extra recordings, is, and if we poke an electrode in a piece of tissue, there are so many neurons that we should not be able to sort the spikes. And we are able to sort the spikes. So most of the neurons do nothing. I don't think they do nothing.
But
we have no clue of what they are doing. That's
yeah, for sure. But so it's like if for example you're considering the lesion. So at the beginning in the 50s, it's true that many behavioral experiments in terms of neurobiology have done with lesion. And now we know that if you remove a part of the brain, we have, I mean, our brain is so plastic that we have a supplementary, we have a substitution of different regions. So you can't really say, okay, this is, this region is really involved in this process just because you remove it, because sometimes it's just another one will take the relay and things like that. I think also the tools evolve, and it's true that we are all invasive. I mean, in biology, I am invasive, and so. Okay, maybe it's not the appropriate word that I should use, but yeah, in the way to implant electrode, it has many consequences, and it's true that, and also for I see with calcium imaging, we are doing fantastic things. But so when you see the chamber that you put on the head of the animals, and so you can record only the cortex, because as soon as you go deeper, you just remove all the brain in the same time. So, yeah.
Okay. So maybe we conclude the discussion for today. I just want to send a message for the young ones who are here. The fact that we are organizing this discussion is also for you, so that you can ask questions to mathematicians or biologists, because even if you have thought about your career and wonder a lot of stuff about, is it true that what they think as a biologist, or, I mean, this is the place where you can also ask stupid questions, and it will be nice that you take this opportunity. It will happen every morning. So it's also for you, and don't hesitate, don't be shy. Today it was a bit slow, but at the end, you see, people were saying stuff that might be interesting for some of you. And if you poke them, maybe you will have your answer. So don't hesitate. And if you're too shy to do it during the discussion, there is also plenty of occasions with coffee, etc., etc. Okay? Okay, so for today, this is this morning, and this afternoon, Daniele will be the chairman. So thank you, Daniele. And so we start again at 2:30. Okay?
I can see your lips moving, but I don't hear any sound. We don't hear any sound here in the...
I'm not able to hear.
So in fact, that's something I wanted. Can you speak again? I would like to ask to... Oh, now the central services are off. So we can't so we can't hear Claire in the room. Yeah. Okay. Can you start sharing your... We can't hear... perhaps I can. Let me see. Perhaps I can from the... Yeah, perhaps I can hear you now from... Claire, can you put up your slides when you were ready? Welcome to this afternoon session. My name is Daniele Vitabile and I am from the Vrije Universiteit Amsterdam, and I'll be chairing this afternoon session on the algebra and geometry. This is the algebra and geometry afternoon. The afternoon is split into two parts. We first are going to have two 30-minute talk by Claire Guerrier and Carina Curto, followed by a break, and then there's going to be a 1 hour joint talk, the algebra and geometry shared by Ida Canari and Catherine Hess. Without further ado, let me introduce Claire Guerrier from Université Côte d'Azur. She's going to be talking about multi-scale modeling of vesicular release at neuronal synapses. Let me remind everyone that I will be collecting questions on the chat on Zoom and on YouTube. And Claire, the floor is yours.
Okay, thank you very much, Daniele. Thank you, Patricia, for inviting me to this conference. I would have loved to be there with you guys, but this year is complicated for everyone. Anyway, it's great that I have the opportunity to come hybrid to present my work. So today, I'm in the algebra and geometry part of the afternoon. So I will focus my talk. I mean, I'm very interested in the role of geometry, how geometry of a cell shapes its function, and how the activity of the cell shapes its geometry. So this is the geometry I'm going to talk about today. Okay, so the first thing I wanted to present is a model for short-term synaptic plasticity. So what would be the motivation for this? We have, in this example, what you observe is two different types of cells, of neurons. On the left, you see the climbing fibers here, and on the right, the parallel fibers. Those two cells are projecting on the same cell, the same neuron, which is called the Purkinje neuron. So the climbing fiber and the parallel fiber are projecting to the Purkinje neuron. They are the geometry of the presynaptic terminal in the two is extremely similar. They have the same size, they have the same number of vesicles, the same maybe organization or number of vesicles ready to release neurotransmitters, but they have very different behaviors in terms of short-term plasticity. So what would be short-term plasticity if you excite the neurons, the climbing fiber, twice within a short time scale? Here you have a 20 millisecond pulse. You will observe in the climbing fiber what we see as depression. So you have, the first impulse that will lead to some vesicular release and some extra postsynaptic current. And then the second excitation, the second pulse, will lead to a smaller pulse. This is called synaptic depression. And if you look at the parallel fiber, it's showing exactly the opposite. It's showing some facilitation. So after the first pulse, you would have a current that will be generated, a postsynaptic current that will be generated. And 20 milliseconds after, the second pulse would be bigger. So the question here is, how come those two synapses that look super similar in terms of geometry and organization and that do project on the same neuron, how come they express those very different behavior? The one is really expressing facilitation and the other one is expressing depression. So this was the motivation of this first study, and to start the model to understand what could be the reason for such discrepancy, maybe I can start with a small background about the functional organization of chemical synapses. So from those of you who are not super familiar with presynaptic terminal, the way it's supposed to work at, let's say, glutamatergic synapses is the arrival of an action potential at the presynaptic terminal. So this is the end of the neuron, the one that is projecting the information to the next one. When the action potential arrives, you have here down at the active zone. This is a specific zone that, as you see, is located in front of the postsynaptic terminal. You have here voltage-gated calcium channels. So those calcium channels would be closed, except if you have a shift in the voltage. So when the action potential arrives, you see a shift in the voltage in the membrane, And this creates the opening of those voltage-gated calcium channels and the entry of calcium ions. Those ions would enter, they would probably diffuse within this terminal. But if enough of those ions are binding on the vesicles that are already attached to the membrane, let's say 4 to 8, it's not yet extremely clear, then this binding of 4 to 8 calcium ions on the vesicles that are already docked at this particular part of the membrane that is called the active zone that is facing the postsynaptic part, then this is going to trigger vesicular fusion. This is the way, like the classical model for vesicular release at presynaptic terminals. Okay, so we wanted to build a model for this and try to understand what could be the parameters triggering such a difference in response to stimulus in short-term plasticity. So if you zoom a bit onto the docked vesicle here, what you observe is that these synaptic vesicles that are docked, attached to the membrane on this region that we call the active zone, which is active where vesicles have to dock, and you have all the machinery and the proteins necessary for synaptic transmission. When you have those vesicles, they are docked to the membrane of the presynaptic terminal through a complex of proteins that is called the snare complex. And this snare complex is related to another protein that is called the synaptotagmin. And this synaptotagmin is the protein trapping the calcium ions. And if enough synaptotagmin has trapped enough calcium ions, then those snare complex are going to change conformation, and then the membrane are going to be fused, and you will get the release of the neurotransmitters that are inside the vesicle. So if you look at this scheme, what you see is that this snare complex is supposed to be located underneath the vesicle, right? It's really those transmembrane proteins, and when the vesicle arrives close to the membrane, then it binds. And you have those calcium channels that are entering also, those calcium ions that are entering through calcium channels somewhere in the membrane. We know that those calcium channels are in the active zone, but what we don't know is how much do they colocalize with vesicles? And this is something that is still not yet understood well in the community. So the idea we had here was that probably reaching the snare complex for a calcium ion is not as straightforward as if the synaptotagmin was like a buffer that was in the middle of the terminal. So we wanted to see how much the geometry of these docked vesicles would interfere into the dynamic of the binding of ions. Oops. So to formalize this problem, we choose to consider the narrow escape problem in a cusp. So which means that we're going to describe the Brownian particles are calcium ions, and we're going to take a look at the first time for these calcium ions, this Brownian particle, to escape our domain here through the red boundary. So the blue would represent the presynaptic terminal, the green would represent the vesicle, and the red would represent the snare complex. Let's say if you're close enough to the snare complex, we consider that you're trapped by the complex of vesicles. So you're going to get mean first passage time in the sense that you have a calcium ion starting somewhere inside this domain, and the Brownian particle starting somewhere, and you want to know how long it takes for this particle to reach this small domain. And this is the first step of triggering vesicular fusion if you have enough of those calcium ions doing this. So the formalism of the narrow escape problem is very good for this question. And we know using Ito's calculus mainly that the mean first passage time U is the solution of the mixed boundary value problem that is described here. The Laplacian U is equal to -1. You have a reflective boundary condition everywhere except at the target site, the red ribbon here, where you have an absorbing condition. So we formalize the problem this way. And then to go quickly on the method, the idea was that if you consider this problem in cylindrical coordinates, you have a symmetry around theta, right? So we can reduce this to a 2D problem. And in this 2D problem, we can now try to think how can we simplify this problem to get the solution? And the idea here was that we were expecting that the time it takes for a Brownian particle to reach, let's say here, to reach the red boundary here would pretty much depend on how far you are on the circles here. That would be all the circles that are tangent in 0. Okay, it's probably not where you are in X or in R or where you are in Z, but probably where you are at least at the first order on the circle. So if you imagine all the circles here, you can probably understand that the mean first passage time to reach the red ribbon would be the same everywhere on those circles, depending on how far you are from the circle. So this idea motivated the use of the conformal mapping method, where we transform all those circles into line. And the idea was that maybe if we get lucky, then at least at the first order, and which is only at the first order, the mean first passage time should not depend on when you are in T, but only on where you are in S. So you transform the problem, at least at the first order, on a 1D problem. So we did a calculation, and using the matching asymptotics, we find that this is true. And then we ended up with a first order ODE to solve, and this is something we're able to do. And we could find some solution for this problem. So here is the solution U of RZ. So you're on symmetrical coordinate, right? And theta it's independent of theta. And this is the kind of solutions you find. So here you find the fact that you are somewhere on the circle, and if you are close enough to the boundary, so when R squared plus Z squared is exactly equal to this boundary, you end up getting 0, which is the time it takes to reach the boundary is going to be 0. And if you fall, you see that this represents where you are on the circle. So we could do this analysis for the 2D domain and the same for the 3D domain. And if you look at this analysis, what you probably recognize is the curve that is here, where you have, when you're far from the boundary layer, you have a constant solution. And when you are close to the boundary layer, then you see a huge drop in the solution that goes from a value that is super high to 0. So this is exactly what we were expecting, because we know that most of the time for the mean first passage time to a small target, the mean time to reach the target, if you're far from a boundary layer, is constant. And then the constant here would be given by in 2D square root of 2r times omega over d square root of epsilon. And in 3D, the omega over 4 pi d square root of epsilon. Okay, so this is the shape of the solution. So what does this result mean? It means that if you're far away from the domain, from the boundary here, this term is going to be extremely small, okay? And this term is going to be the leading order term in square root of epsilon. So the time it takes for a Brownian particle that starts somewhere outside of the boundary layer is constant. This is what you see here, and is given by this formula. Epsilon is the size of the absorbing window. The 3D domain is the same. Sorry, there is no square root here. This is a mistake. This is 4 pi d epsilon. We could check this versus Brownian simulation, and it gives excellent results, as you can see. Okay, so we had this idea. Maybe the first thing is that we could compare these to other mean first passage time in other geometries. So actually, what we understood here is that like the time it takes is in 1 over epsilon, where the surface of the hole is in epsilon at the power 3.3 over 2. So the surface of the hole would be the surface of the small ribbon. You had already known results, like, for example, if you want to try to reach a small hole in a sphere, where the sphere is of radius a, then this is the mean first passage time you would get, where the surface of the hole would be like pi a squared. This is just a circle. You can do the same, like the results in the funnel shape curves here has been also derived. This is something that you can use probably when you do like meiosis and mitosis for the cell division. This is a kind of geometry you can find. And again, the mean first passage time, it has a different dependence in the size of the hole. So what if we put everything together here, so what we get is that the mean first passage time from a small hole at the first order is constant. Actually, what has been shown is more than that. It's that the law of arrival is exponential at the first order. Okay, so what it means is that this mean first passage time is integrating the effect of the geometry in the rate, but you can then consider using this rate that what you have is the arrival of your particle is a Poisson process. So this is a way to integrate a very complex geometry into a very simple tool, very simple model. Okay, so that you can consider the arrival at the first order, the mean first passage time is independent of the position if you stop outside of the boundary layer. You know that the law that is associated to it, this is something that has been shown, is exponential. So what you get in the end is a rate that captures completely the geometry and that gives you a Poisson process for the arrival time. So this is the most important and most interesting probably results you have with this mean first passage time at small holes when you're trying to do modeling and multi-scale modeling. Okay, but if you we were interested in the model of the full terminal, so what we know now is how long it takes for a calcium that is far away to come back. Okay, but what we don't know is what's happening at the active zone. So let's say the active zone usually model it this way, okay, with several vesicles on a square lattice, and the question of where are the active the channels is a question that is always under debate, and it's probably different depending on what kind of synapses you're looking at. So the thing is, and this drawing is maybe easier to understand. The thing is, our calcium ions is entering inside the boundary layer, actually, or might enter inside the boundary layer. So now that we resolve what's happening outside, we have to also try to resolve what's happening inside the boundary layer. So we have already the shape of the solution inside the boundary layer, at least at the first order. But what we're interested in is now the splitting probability. So it means like when you enter the boundary layer, when you start somewhere inside the boundary layer, what is the probability to reach the target before getting lost inside the terminal? And this is called the splitting probability. And actually the PDE that is solved by this problem is kind of close to the one we had before. So using the previous analysis, we could find an approximation of this probability. So we found that the probability takes this form, and we could estimate A using Brunel simulation, and we end up with the approximation of the splitting probability being this one. Okay, so this is the probability to enter the boundary layer before getting lost inside the terminal. So now if we want to integrate all of these things in a full model, the main problem we're going to get is that inside the bulk here, what we have is a classical dynamic of calcium binding on buffers and pumps and whatever. And this classical dynamics is going to be represented well by deterministic equations, right? This is let's say the mass action laws to be super simple. Whereas here, the entry of calcium inside the active zone through the voltage-gated calcium channels and the binding or not binding on the vesicles is well represented by a stochastic approach. So we choose to couple the stochastic approach to the continuum through this Markov model coupled to mass action. So the idea was to represent with continuous equation the bulk, and with a Markov chain the target, the binding to the target, the discrete and the stochastic part. So, and we could do this thanks to the mean first passage time theory and thanks to this arrival rate of the Brownian particle to this snare. That's thanks to the fact that this is an exponential low, then that we could represent this as a Poisson process. Okay, so then you do the simulations, and you can observe the distribution of release time or uniform channel distribution. So what you can see, I did different simulations for different number of buffers, okay, that bind the calcium. So for a different shape of the calcium impulse curve. And what we could see is that this calcium shape is actually triggering the vesicular fusion. So after calcium entry, you would see a huge peak corresponding to vesicular release. And then if the calcium stays high in the cell, what you would see is a lot of vesicular fusion happening after that due to the ions coming from the bulk to the active zone. If you lower down the calcium, you will lower down this second peak. And if you don't have any calcium, because you have a lot of extrusion or buffers, then you would not see this peak, okay, this vesicular fusion. So this was a first hint into what's happening in the synapse and how controlling the level of calcium inside the cell, which is something that is extremely complex, and that the cell is paying a lot of energy doing it, can control also vesicular release. So a lot of one of the main idea of this is one of the main idea of facilitation is that it's coming from higher calcium inside the terminal. So it's not exactly the way it's been described, but what we see is that this is true, that the higher calcium in the terminal is going to trigger a lot of vesicular release inside the, like, not that directly because the calcium is high, but because you have all this process of coming back and probably like synaptic priming, et cetera, et cetera. So what we wanted to investigate at the beginning, and this is probably, I will not have time to go further than that, is that what we could see by changing some parameters in our full model is also that depending on how you could, like on some properties of your SNAP complex, so the complex of protein that is attaching the vesicle to the membrane, depending on the dynamic of this complex that is not yet well known and it's under debate, you could, with the same, calcium, let's say, calcium dynamic, obtain very different release time. Okay, so this was a way to investigate how fast snare unbinding and slow snare unbinding could affect vesicular release. And what we could see is that if you have a slow snare unbinding, so the calcium can unbind for the snare, but slowly, like depending on the buffer, the number of buffers you have inside the terminal is able to make the synapse shift from a facilitating synapse, which you see here with the purpose ratio that is bigger than 1, to a depressing synapse that you see where the purpose ratio is smaller than 1. So yeah, I have other things, but probably I don't have time for it. I don't know, Daniele,
you do
but
have a few more minutes, given that we also started a bit later, so you can take.
Okay, maybe let's stop here, and if people have questions, I would be happy to find it to answer.
At the end, talk then. Claire, can you hear me?
Yeah.
Is that the end of your talk, or do you want to?
Yeah.
Okay.
Yeah.
Well, thank you very much then. For a change, let me start from the questions online. So we have a question from Romain Vels. Romain, just perhaps unmute yourself and ask the question yourself.
Hello, can you hear me?
Yes, we can.
Yeah.
Thank you very much. Thank you very much for the talk, Claire. It was very nice. I have two questions. The first one is, can you discuss the modeling of ion channel dynamic of calcium dynamics by a Brownian motion? Do you think it's plausible? And for example, do you have any interaction between the ions, or if you have too many in a specific, for example, if the density is too high, or if you have actin and preventing the free the free movement of the ions. Do you have any idea?
So it's a huge assumption. Anyway, the vesicle is never that crowded so that the motion of ions is perturbed, I think, a lot, so that the Brownian motion is not still a good result. So usually people, they measure the diffusion coefficient, and this measure of the diffusion coefficient is already taking into account of kind of crowding, and we are not in the case where you have so much ions that the crowding prevent them for having this almost Brownian motion. The other thing is that it's more problematic when you reach calcium sensor, for example, when you reach the synaptotagmin, because then you know that you have this complex of protein that is charged and that is kind of attracting the ions. So I feel for me the biggest problem would be when you're reaching the snare or whatever buffer or pumps. So the way in modeling we resolve this problem, for example, for the buffers, actually the radius, actual radius of your buffer, and if you think about Smolachowski, is the radius that takes into account the tracking.
Okay.
But you get kind of a bigger radius than the real ones, because when you measure this Brownian rate, actually the fact that when you're close, you can trapped is already taken into account. That's why we choose to have this ribbon instead of having specific sites underneath, saying like, okay, if you're close enough to this snare complex, what's happening is probably too complex to be described with simple Brownian dynamics, and we don't really want to enter into these details. So we just let go. The other thing is that we know with the Poisson-Nernst-Blanc theory that the charges are pushed on the boundaries, so we probably ignore a part of these Brownian ions that will be directly pushed on the boundaries. But the center of the cell is supposed to be electroneutral. So Brownian motion doesn't seem too bad.
Okay.
Yeah.
Thank you.
Bruno, you have a question?
Yes.
Go ahead.
Can you hear me?
Yes, we can.
Yeah.
Hello, Claire. Yes, my question is about the beginning of your talk. You were assuming that the spike is arriving in order to trigger the whole synaptic plasticity process. But as far as I understand, you don't need a spike. You just need to have a rise in the voltage. I am asking that because not all neurons are spiking. In the retina, most neurons are not spiking, and it is believed that short-term plasticity could arise also in the retina. So would it be possible to adapt your model in the case where you don't have a presynaptic spike, but just a rise in the voltage?
Yeah, sure. So I took the standard description because this is a very general model. Yeah. And the idea here is that the molecular machinery I choose to let the calcium enter is the voltage-gated calcium channels. Yeah. And they have a pretty high voltage shift. But the voltage-gated calcium channels, they don't need an action potential. They just need a shift that is high enough so that they open. If you have different types of channels, low voltage channels, whatever kind of calcium channels, you can get exactly the same kind of results. The idea is just having calcium entering. So yeah, it's true that I simplified a bit by saying like action potential rise and the voltage-gated calcium channels enter, but this model doesn't care that much about the action potential. It's just about representing the flux of ions entering.
Yeah, thank you. Thank you very much.
Thank you. So we have no more questions online, so perhaps we have questions in person from here.
Yeah, thank you for your talk. I'm wondering, first slide you showed we had one case with a climbing fiber exhibiting paired-pulse depression and the parallel fiber paired-pulse facilitation. And at the end, you just explain, so to speak, facilitation. And if I get your model right, facilitation is due to saturation of endogenous buffers, so that when the next calcium entry comes, you have more free calcium available or not?
Yeah, I think this is the classical idea, and I didn't like it. I didn't trust it as a student because it didn't make any sense for me that the fact that you have more calcium inside the terminal would lead to, when the second spike arrives, bigger release. Because I was like, the time scales are not the good ones. Like the time it takes for one ion to find back with this multi-expression time, it's giving something like 4 seconds. Is it 4? Is it 3? Is it 2? Who cares? It's orders of the seconds, right? Whereas the voltage, the fusion of vesicles is less than milliseconds. It's microseconds actually. So it didn't make sense for me that the fact that you have more calcium inside the terminal would lead to bigger release. So, but this is what I've been observed experimentally. And with this model, I find an explanation that at least I understand and that makes sense, and that some people call like snare priming. So actually, when the calcium is high, you have some of those calcium. So this is Poisson process. So the more calcium you have, the smaller the time it takes for the first one to arrive, right? You divide by n the number of ions. So actually, if the number of calcium is high, you would get some calcium ions coming to the snare. Because you need 4 to 8, you can imagine that you have a first spike, and so you release the vesicles that are close to the calcium channels, and you release the first vesicle. Some vesicles are not released. Some calcium ions are still coming back because calcium is high, so you still have this calcium coming from the bulk, priming the snare. Let's say you have 3, 4, 5 calcium ions that are left from the first spike and from the fact that you have this arrival because calcium is high. Then when the next second spike arrives, you don't need 8 anymore to activate this snare. You need only 3. And that's probably why the probability to release is getting higher. So this was my understanding of what could cause this, the fact that when you have a higher calcium, you're going to get a higher release. This is something that's been observed. So, but this is, and because the unbinding from the snare is not something that is really well known, if you have a very fast unbinding, this is not going to be true. But if you have a slow unbinding, this is something you're going to observe. This was my point, I think, in this. I would have to test this, but I think people have done some stuff in the literature, but yet, it's something we should see with people doing experiments. But my idea would be this one, that you can shift due to this calcium.
Thank you.
Any more quick questions? We have perhaps time for one. If not, let's thank Claire for a very nice talk.
Can you hear me?
Okay, our next speaker is Karina Curtu from the Pennsylvania State University, and she's going to be talking about graph rules and topological insights for inhibitory network dynamics. Karina.
All right. Thank you very much. There's a little bit of feedback, I think, when I'm talking. Can that be fixed? Sorry. Okay. Maybe it's the microphone. All right, I'll try. I'll try not to hurt your ears too much. It's there, yeah. Should I?
We're just waiting. There is a glitch in the audio system.
Yeah. All right. Should I keep talking so we can see what's going on? I think it's better. Yeah. All right. I'll get started. So I have 30 minutes. All right. So anyway, I'm pleased to be here. I'd like to thank the organizers. It's very convenient to be able to come to this conference because I have a collaborator at ENS just down the street. So it's very nice to be able to finally, after 2 years, take a trip here to Paris. Okay, so let's get started. I want to begin by thanking my collaborators. So first and foremost, I'd like to thank Katie Morrison, who has really worked on me for all of this work that I'm going to, almost all of it that I'm going to show you today. She's a professor at the University of Northern Colorado and just a long-term collaborator of mine. Caitlin Parmalee is a former graduate student, and Chris Langdon and Jesse Genison were former postdocs in the lab. Nikki Sanderson, who's here, is a current postdoc in my group. And then Josh, Juliana, and Caitlin are current grad students who have also worked a lot on this work. And you'll see some work of Juliana's, especially towards the end. And I'd also like to thank Vladimir Itzkov and Anna Degeratu, who did a lot of work in some of the precursors to what I'm going to talk about today. And finally, my WinComp group, who I probably will run out of time to get to the nerve theorem stuff that we did together, but just in case, I'd like to thank them always anyway. Okay, so let's get started. The starting point, sort of conceptually, for what I'm going to talk about really goes back to the 80s with the work on the Hopfield model and Hopfield networks. So this should be familiar to most people who study mathematical or theoretical neuroscience, and many neuroscientists more widely, of course. And the basic idea in these models is that you're going to model memory patterns or stored patterns of activity in a network as fixed point attractors of the dynamics. And this really, kicked off this idea of attractor neural networks. So these ideas were around before, but the Hopfield model really made them popular and brought a lot of physicists into neuroscience to study questions such as, how many attractors can you store in a network, capacity questions, and various aspects of how neurons can compute using this kind of framework, right? So this is very much the framework of a recurrent neural network that has attractors. And in my work, we are focused on not the Hopfield model, but sort of I think of a more modern version of it, threshold linear networks. And these are really a firing rate model. So again, you have neurons, and they're interacting according to recurrent connections. So there's a W matrix that is capturing the strengths of the connectivity. And the dynamics of the neurons really are evolving according to the so-called threshold linear dynamics. So there's a leak term. In the absence of recurrent inputs, a neuron will just decay to rest or to a zero firing state. And then the recurrent inputs are captured by this sum of the Wij, Xj. And there's a combination of threshold and external input that's captured by the Bi. And so we're going to refer to this, the data that kind of specifies a particular threshold linear network is really the pair, the matrix W and this vector B. okay? And just this notation, the brackets plus, is denoting the threshold nonlinearity. So it's a maximum between 0 and the argument of the function, okay? So the machine learning people like to call that ReLU, or the rectified linear unit. So perhaps remarkably, these networks can display the full repertoire of nonlinear dynamics, even though you may notice already, if you think about the equations a little bit, locally, these are linear systems of ODEs. So what's happening here is I have a piecewise linear system of ODEs. So the equations inside this nonlinearity can be thought of when you set that to 0, which is when you switch from one side to the other of the nonlinearity. That defines a hyperplane in the state space. And each one of these equations has its own hyperplane with the normal vector given by the row of the W matrix and a shift given by the B sub I term. And this hyperplane arrangement chops up the space so that locally you have honest-to-goodness linear system of ODEs, because you've picked one side or the other of the nonlinearity in each chamber. But despite that, it's in the trajectories passing between different chambers that you see all the nonlinear behavior emerge. Okay, and this can arise even for effectively inhibitory W matrix. So, 20 years ago, I would say the kind of mathematical study of these networks really kicked off with some beautiful work by Hanlesser, Sung, and others at MIT at the time on the symmetric case. So they were studying threshold linear networks where the W matrix is symmetric. And in that case, you can prove a result very similar to the classic Hopfield result, where you can guarantee convergence to one of multiple stable fixed points. And with Anda and Vladimir, we followed up on this and continued working on the symmetric case for a while. So we wrote some I think, interesting papers about the collections of fixed points that you can have and what kind of neural code you can encode. Then I started working with Katie, following up on that. But really, we wanted to get away from the symmetric case. So we started there because that was in some sense simpler to tackle. But today, I really want to talk about both. I really want to talk about non-symmetric W matrices. And the focus now is not just on stable fixed points and fixed point attractors, but on stable and unstable fixed points, and because that enables us to look at dynamic attractors. Okay, so this is just kind of a dump of references that where you can find things that I'm talking about today. But I want to sort of get straight into the motivating question, which is what is the relationship between the attractors of a network and its recurrent network architecture? And the focus is really on the pattern of connectivity as reflected in a directed graph. Okay, so we really want to answer the question, suppose you're given the structure of the network, suppose you're given a directed graph that captures that structure, what can you say about the attractors? And I want to make this very clear. We care not just about stable fixed point attractors, but also dynamic attractors, limit cycles, quasi-periodic attractors, and so on. And we want to be able to read that off of the structure of the graph. And that requires us to look at both stable and unstable fixed points. So let me give you an example of the kind of rich dynamics that you see in these networks. So here's a network with just 9 nodes. So I've shown you the graph of it. This graph is giving me a W matrix, which I'm not yet showing you, but let's just say from this graph I get a W matrix. And then I can evolve the network according to the TLN dynamics. So remember, these are just the dynamics that I showed on the previous slide. Okay? So there's a W matrix coming from that graph. And now we really want to focus on the effect of the local connectivity on the dynamics and not think of the dynamics as being reflecting a changing external input. So we're setting the external input parameter, the B, constant. Okay? So every neuron is getting exactly the same external drive. And so the different dynamics that you see, this is I'm falling into a stable fixed point where a pair of nodes 4 and 8 are firing. A different initial condition takes me to a different stable fixed point where 1, 8, and 9 are firing. Note that there can be overlap between the neurons that are involved in these different attractors. So these are the classic Hopfield style stable fixed point attractors. But now when I go on to some other initial conditions, I can fall into a limit cycle or even a strange or chaotic attractor. okay? Again, for different initial conditions. And all of these attractors, the remarkable thing about these networks is that all of these attractors are in some sense predictable from the structure of the graph, okay? So how do we get to the W matrix from the graph? So we consider a special case of threshold linear networks. In order to really focus in on the graph structure, we consider a special case where the graph is fully determining the W matrix. And, but we are staying within the realm of inhibition-dominated networks. And so what we imagine is that we have a network of excitatory neurons in a sea of inhibition. So there's some strong inhibitory background network, or a global inhibition, if you will, that is causing pairs of neurons to effectively inhibit each other even when there's an excitatory connection between them. So you think of the total interaction as being a sum of excitation on top of this strong sea of inhibition. And this yields to two values for the interaction strength. So there's a value -1 plus epsilon. Epsilon need not be small, by the way, but we just chose epsilon and delta as our two parameters because we had used other Greek letters, too many other Greek letters for other things. And this is a value above -1. The -1 minus delta is a value below -1. Where does the -1 come from? The -1 is the self-interaction term, and it is the strength of the self-inhibition in the absence of recurrent inputs that are superthreshold, okay? And I should note as well that there's an implicit time scale here for the leak that has been set to 1. And so it's not that there's something special about 1. Really, the -1 is in units of this time scale that we've set to 1. And so what we're really looking at here with these W's is when there's an edge in the graph from I to j, or from j to I, then Wij is bigger than -1 because that means that j inhibits I less than it inhibits itself through its own self-leak term. When there's not an edge, the inhibition is stronger, so more negative than the -1. And so you really should think about the graph as encoding the pattern of weak and strong inhibition. Okay, when there's an edge, it's a weaker inhibition. When there's not an edge, there's the strong, just the background strong inhibition. And weak versus strong is determined with the reference point, which is the self-inhibition from the leak term. And so we think of these as generalized winner-take-all networks, but you can just think of it as a special case of TLN dynamics. So if we wanted to sample different networks and try to look for interesting rich structure, instead of just taking random matrices, random W matrices where we're perturbing around some point, this allows us to use these graphs to find kind of faraway points in the parameter space where interesting differences in the dynamics arise, and to connect those directly to the structure of the graph. Okay, so I'll try to speed up a little bit now, because I want to get to a cool application at the end. But one of the things you can notice right away with these networks is that you really can tackle the different emergent dynamics fully as sort of purely as a function of the structure, of the global structure of the graph. So I'll just quickly say, here we have 4 different networks. This is a kind of a simple example to show the idea that the local degree sequence is not enough to sort of determine the dynamics in these networks. So all 4 of these networks are degree matched. That means that if I look at the in degree and out degree associated to each node, so for instance, node 1 in graph A here has in degree 1 and out degree 2. So I give it a 1, 2. I do that for all the nodes, I get the degree sequence. And I do that for every graph, and every graph has its own degree sequence. And I sort it in some canonical way. All of these graphs are identical as far as their degree sequence. They're also identical, the networks that I've made from these graphs are identical as far as the epsilon and delta choice. Okay, and so everything locally is the same for all of these networks. The only difference is the actual structure of the graph. Okay, and so if we see different dynamics here, right, we can truly say this is emergent from the connectivity structure. It's not due to changing external inputs. It's not due to anything other than changes in the connectivity structure. And there are wildly different dynamics, okay, in these degree matched graphs. The first one has a global attractor, a single limit cycle that always emerges no matter what the initial condition is, okay? And it corresponds to a 3 cycle in the graph, 2, 3, 5. And you can see that actually in the limit cycle, the activity follows the arrows of the graph. Okay? In the second case, we get 2 limit cycles. In the third case, we get 3 stable fixed points. In the fourth case, we get 4 chaotic attractors. So all of these are quite different. But of course, we've seen in the previous example that you can also have these different kinds of attractors coexist in the same network. Okay? We also get some very interesting dynamics in highly symmetric cases. So here I have what we call the 5-star graph. So it's a tournament, and it has a cyclic symmetry. There's a boring, I call this a boring attractor that's associated to it. It's a limit cycle where the neurons are where the activity is following the edges along the outside of the graph. So 1 to 2 to 3 to 4 to 5, and so on. And you can see the rate curves are color-coded according to the neurons. So these are the curves xi of t for each of these neurons. Now, the exact same network, exact same W matrix, different initial condition. All I'm changing is the initial condition can now fall into this completely different attractor, which I call the Gaudí attractor because it reminds me of the architect in Barcelona. And I've never, prior to this, I'd never seen anything like it. The curves look like they're melting, okay? But this really is an attractor of the network, and it's very stable and reproducible. And it corresponds to to a different cycle in the graph, 1 to 3 to 5 to 2 to 4 to 1. Okay, so you're seeing now. Let me just take a moment to remind you. These are effectively inhibitory networks. Every neuron is actually inhibiting every other neuron. Okay, so it's not like we're flowing along edges of the graph. The edges of the graph are indicating weak inhibition. The non-edges are indicating strong inhibition. But it's not too bad of an idea to think of flow along the edges, because in fact that's what happens, right, when we look at the dynamics. So that's what makes this sort of a nice graph. And then here, let me just show you an example in case for those of you who know about quasi-periodic attractors. For this other graph, we also see 2 different limit cycles. Well, sorry, 2 different dynamic attractors emerge. The first is a limit cycle, kind of the boring one, going along the outside. And the second is a quasi-periodic attractor. And what you see here is a projection of this beautiful torus that's traced out in the 7-dimensional state space. And we've just taken a random projection so that you can see the torus structure. Okay, so how do we analyze these things mathematically? We look for the fixed points. Okay, so obviously, fixed point attractor, the stable fixed points correspond to attractors. They are their own attractor. But it turns out that the unstable fixed points are also extremely useful for figuring out dynamic attractors, okay, in these networks. And the way we study the fixed points is really by thinking of this geometric picture of the threshold linear networks as this patchwork of linear systems. So I said a little bit about this in the beginning, and let me say it again. You can think of your n equations, right, as having, as defining a hyperplane arrangement. So the argument inside the nonlinearity, we refer to this like y1 through yn, and setting yi equals 0 defines a hyperplane in my n-dimensional state space. And so I have n hyperplanes, 1 for each equation. And once I'm inside one of the chambers of this hyperplane arrangement, I have picked one side or the other of the nonlinearity. So either I can drop the brackets because I'm on the plus side, or I set it to 0 if I'm on the minus side. And now I have an honest-to-goodness linear system of ODEs inside that chamber, right? So you have all these different linear systems. They're related to each other because they're all coming from the same W matrix, but locally the system is linear. And there are some nice things you can unpack from that. In particular, there's a one-to-one correspondence between the fixed points of the full TLN and allowed supports of fixed points. So it turns out that in order to have a fixed point of the full network, what you need is to have a fixed point of one of those linear systems that actually lies inside the correct chamber where that linear system is governing the dynamics. Right? Every one of these linear systems is, we have a non-degeneracy condition actually that we require for our W matrix. So every one of those linear systems has a single fixed point associated to it, but that fixed point may or may not reside inside the chamber where the linear system is governing the dynamics. Okay, and only if it does is it a fixed point of the entire system. And so the whole game is to figure out, okay, which chambers contain their own fixed points. And so you can really label the fixed points by their supports, which are subsets of the neurons 1 through n. And that is the game. And so this fp of w or fp of g, when we think of a particular graph with epsilon and delta parameters, is telling me all the allowed supports. And so you can, here's a very simple case with a 3-cycle. You see this nice limit cycle attractor that we get. Here's the hyperplane arrangement with the different yi is equal 0. There are 8 chambers that you're chopped into. And the limit cycle necessarily has to cut across more than one chamber. It cannot reside inside a single one because that's a linear system. Linear systems don't have limit cycles. And so the limit cycle is actually emerging as a trajectory that spirals out from that central chamber and then cuts across different chambers, but like comes back to the central chamber and catches the spiral at an earlier point. So it's always spiraling out when it's in the main one. And then this patches together to give a perfect limit cycle. Okay, so, all right, so hopefully I've convinced you at this point that we care about the fixed points and that there's a combinatorial flavor to them and that we can think of them nicely and productively in terms of this hyperplane arrangement, in terms of this geometric picture. So now I just want to say very quickly that from these insights, we're able to develop a theory of graph rules where we can actually analyze the structure of the graph and know something about the fixed points, right? So we can prove theorems that say if the graph has this structure, there's going to be a fixed point with this support. Or if the graph has this other structure, there will not be a fixed point with this support. Okay, so the graph rules are ways of looking at the combinatorial data in the graph and predicting fixed points or predicting absence of fixed points. And so I will just show you one of them, one of the simple ones, which is the uniform in degree graph rule. And that one tells you that if you have a subgraph that is uniform in degree, which means that every node is receiving the same number of incoming edges. So up here are some examples. They need not all be symmetric. So for instance, graph A4 is uniform in degree 1. Every node gets only one input. These graphs are special in the following sense. So if you have in your larger network an induced subgraph that is uniform in degree, then you're guaranteed that the nodes in that subgraph support a fixed point. Meaning there is a fixed point where only those nodes are firing and all the other ones are silent, if and only if there's no node outside of the subgraph that receives d plus 1 or more edges from the nodes inside. So sigma is the collection of nodes inside. And d is the degree, the uniform in degree. So from this theorem, I can already answer sort of a question you might have had earlier when you looked at this example. We have this nice limit cycle corresponding to the 2, 3, 5 3-cycle in the graph. But there's another 3-cycle in the graph, 1, 4, 5, and there's no corresponding limit cycle for that one. What's different? What's different is that one of them has a surviving fixed point, the other one does not. So there's an unstable fixed point associated to 2, 3, 5, okay, which is kind of at the heart of this dynamic attractor of the limit cycle. It kind of goes around it. But 1, 4, 5, there is this external node 2 that receives too many edges from it, and that kills the fixed point. So the graph rule is telling me which of these subgraphs is going to keep its fixed point when embedded into the larger network. And the ones that keep their fixed point are the ones that carry their attractors with them. So that is the heuristic that we use. So we can prove things about the fixed points, and then we have a heuristic that tells us how to connect certain unstable fixed points to predictions about dynamic attractors. Okay, so we have many of these graph rules, and I don't have time to get into it. Now I just want to, I have 5 minutes left maybe? Okay, because I started late. Now I want to show you an application, okay? So, to make a long story short, we've been able to prove a variety of rules like this. And in fact, we're even now working on sort of axiomatizing them in the following sense. So we have many, many graph rules, but we can strip down to some elementary ones. So I'll just show them. There's some elementary graph rules that we prove directly from the TLN equations, and then sort of derived graph rules that we can get once we know that these are true. Okay, so we're trying to really systematize the graph rules and figure out like what's sort of the essential ones from which we can prove subsequent ones. And now I want to tell you about one of the more complicated graph rules that stems from the elementary ones, but that leads to this nice application. And that is graph rules about simply embedded graphs. And in particular, as a particular application, we have this concept of the cyclic union. So if you take a bunch of graphs and connect them together in a way where the nodes from one of them will all have forward edges onto the nodes from the next one in a cyclic manner. So tau 1 through tau n are the collections of nodes corresponding to graphs 1 through n. And now I collect them in the cyclic union. Then it turns out that I can prove a graph, well, we've proven a graph rule that tells us the fixed points of the large composite network from the fixed points of the individual ones. Okay? And what it says is that precisely that the fixed points of the larger network emerge by gluing together supports for fixed points from the individual subnetworks. Okay? If you think about the hyperplane arrangement picture, it's kind of crazy because you have these hyperplane arrangements for the individual subnetworks. Now you sort of glue them together into this giant hyperplane arrangement. And now I'm telling you that the fixed points of the large one are completely determined and predicted in a very simple way by knowing the fixed points from the smaller networks and just gluing them together in all the different configurations. Okay? So this is a nice rule, and there are many related rules like that follow from some of these elementary graph rules. The gluing idea, I think, is very nice. And for those of you who know a little bit of topology, you almost have like this pre-sheaf structure where you think of the fixed points of the subgraphs as kind of sections of a sheaf. And then the gluing is sort of like a sheaf property that allows you to take the local sections and glue them together into fixed points of the whole network. So there's some nice structure there that I don't really have time to talk about. But I want to get now to the application. So using the cyclic unions graph rule, which Katie and I developed in collaboration with my former grad student, Caitlin Parmalee, we are able to engineer some very nice networks with prescribed attractors. And this is work of my current grad student, Juliana Londono Alvarez. And she was able to tackle this question of having simultaneous limit cycles encoded in the same network to reflect different quadruped gates. Okay? So this is kind of an old problem in neuroscience of central pattern generator circuits, is you have animals that can support different gaits in their locomotion. So the classic example is you have a horse, it can walk, it can trot, gallop, pace, etc. It has these different gaits. Pronk is actually not one that the horse can do, but we can put it in the same network. So we just, it's like a super animal that we've designed this for. And if you look in the literature, even very recent literature, like this Nature Communications paper from 2019, most of like all of the models really require parameter changes to go from one gate to another. Okay, so they posit synaptic plasticity or some kind of gating variable has to change or some kind of parameter change to the model in order to transition from, say, walking to trotting. Okay? And we wanted, using the machinery of our threshold linear networks and using our graph rules, we wanted to see, can we make a network where all of those limit cycles for the different gates coexist without having to change parameters. That it's just a matter of initial conditions, whether you fall into one gate or the other. Okay, so in principle, it's very difficult to do because you have these different limit cycles that have the feet, the same neurons that control the leg motion, going in different patterns, and you want to create a circuit that allows for all the different patterns without changing any parameter, truly coexistence of those patterns of activity, just from it and accessing one or the other just based on changes of initial condition. So Juliana set to work, and using the cyclic union idea, she was able to make circuits, okay, for different gates. So first she did one at a time. So here's her circuit for the bound gate. Here's her circuit for pace. So the pattern of the feet hitting the ground or the hooves hitting the ground is different in each case, but she's got to be reusing the same neurons, the left front, left back, right back. Okay, so these colored ones are the different neurons that are controlling the legs. Then she did trot. That one's different because they have to hit in a very different pattern. Walk and pronk. Pronk is a very cute one where they're all up at once and then come back down. The horses don't really do that, but we wanted our model to sort of do even more. Okay, and so for each of these, using the cyclic union idea, she's able to get them to have the right pattern of activity. So you can see these graphs at the bottom. I'm showing the pattern of activity. You can see it emerges in the network. Okay, so she gets it to work. And then here's the miracle. How do you make a network that has all of them? We glued these together. We literally just took the union of the edges in all of these graphs, okay, and just identified the nodes that were for the same feet and glued it all together and made a super graph, okay? And now the question is, do we have any chance of preserving those attractors that she carefully designed, for these subnetworks? And so this is the super network. It has 24 nodes. There's the same 4 that are, many edges coming in and out of them. It looks very complicated, but it's really just a gluing of those simple networks that she had, one for every gate. And this is the adjacency matrix. And now I just want to stress for you, this is again in the framework of the combinatorial threshold linear networks. The only data specifying this network is this adjacency matrix, the epsilon and the delta. And that tells you what the W entries are. So that gives you the full W matrix. And then theta is set to 1. Okay? So all the information to make this network is really here on this slide. Okay? If you can read all the edges off of that graph. It's a little bit of a messy picture. Okay? And lo and behold, from the super network, we see all of the individual limit cycles. They are all preserved when we glued it together. Okay? That actually we did not have theory to guarantee that would happen, right? But it's just these different solutions are kept so well separated, right? Somehow the network is able to maintain them all. And this means, by the way, that because they're all there, they're all stored in the network, you can go from one gate to another by a simple kick. So here is now a simulation with one initial condition, and we started in the bound gate. And then after a while, we kick neuron 15, which is one of the neurons involved in the pace gate. It's not one of the 4 legs. It's one of the neurons involved in one of those cyclic unions, but not in the bound one. We kick that neuron. That just means we make the theta value for that neuron go high for a little bit and then come back down. So then we're back to exactly the same network in this period. And now we're in pace. And then we kick another neuron and we transition to walk. And another neuron and we transition to prunk. And another neuron and we transition to trot. And so you can see here, because these all coexist in the same network, we don't need any change of parameters. Nothing has to change about the network. All you have to do is kick a neuron to sort of take you to another part of another basin of attraction, and then you fall into the other gate. And it's very, very simple, right? So if you had to design animals, right, and how they would then locomotion for them, this would be a very elegant solution, right? You don't need to have synaptic plasticity or any strange changes to the network. You just naturally transition from one gate to another. Okay, now I think I am finally out of time. We added noise to test for robustness. So I just flash, this slide about the noise. If we add 1% noise to the W matrix, we still see the perfect transitions. If we kick up the noise, we see some transitions fail, but maybe we just have to stimulate more strongly and so on. And so there's a lot of tests we can do for how robust this is. But these limit cycles really are, they're coexisting in the same network. Okay, and I think I will end there. This is the last bit with the Wincomp group, the nerve theorems, where we use this. We can cover our large networks by these directional graphs that are special. And this allows us to reduce to the nerve of the network and have a subgraph that is simpler. And so we we've been able to prove some theorems, some nice theorems connecting the fixed points of the large network to the fixed points of this reduced graph. It's like a dimensional reduction technique, but with an eye towards the fixed points. And that also allows us to predict dynamics and so on. So I will end here, my conclusions. First of all the fixed points matter. By paying attention to both the stable and the unstable fixed points, we're able to get a handle on dynamic attractors. The graph structure of the TLN is heavily constraining the allowable dynamics. So this really is a model where you have a chance. It's surprisingly tractable. You can analyze the graph and make predictions about the dynamics, and then run the simulation, and it works. And then I talked about this application with the CPGs. So just I want to thank again my collaborators, and then maybe I'll end with this slide. We are having a semester-long program in a couple of years at ICERM, which is one of the math and computation institutes at Brown University in the US, so it's NSF funded. And we recently got approval to run a full semester-long program. Actually, one of the co-organizers is Catherine Huss, who's talking next. And this is going to be a mix of people with backgrounds in mathematics, in particular applied topology, and theoretical neuroscientists. And so the goal of the program is to really bring mathematicians and theoretical neuroscientists together to work on developing mathematics that will push the fields forward. So if you're interested, shoot me an email, or you can Google this and find the program. But we are now starting to put together the workshops, invite people, and so on. And we would love to have people in this group apply or show interest, and we can invite you, and so on. So please. Anyway, just advertising. It's so awkward to advertise stuff, but there it is. Okay, I'll end there. Thank you so much.
Any questions from the in-person audience? Patricia.
Thank you for this very nice talk. I'm not a specialist of dynamical systems, so at one point you said, so you're sure you created a graph that has all the fixed points that you wanted. Mm-hmm. And then you said you still have to check that all the rhythms coexist. Why is it not sure once you have the theoretical?
Oh, so the theoretical results do not tell us. We have heuristics, we have patterns that we see to guess the dynamic attractors, but just knowing the fixed points does not guarantee for us what the dynamic attractors are going to be.
Okay.
Yeah. So the limits, even the existence of these limit cycles is hard to prove, right? Okay. And so, but we know they're tightly connected. So it's like because of that heuristic, right? Then once we prove things about the fixed points, we're like, oh, we believe now, and then it's like more of a theory in the science sense, right? Okay. Where it's like we can predict them, but we still have to run the simulations to see that it actually works. Mm-hmm.
And also, I had a more maybe philosophical question. Mm-hmm. You said that this is linked. This could be seen as linked to artificial neural networks because this is a ReLU function or whatever. You see?
I actually do not stress that, but.
Well, we could imagine a little bit.
Uh-huh.
And the way, for the moment, artificial network works, I think it's the fixed points that they are working on. Like, you give an entry, you go until the end, it gives you this answer. And the way you're presenting it, it seems that you could encode much more with less neurons by also looking at all the rhythms that you can create with all of that. Mm-hmm. So what would you think about that?
Yeah. I mean,
no, but
I think
could
deep
you
learning
try
networks,
to design something?
I mean, I think the classic deep learning networks, right? It's really implementing an input-output function. That's it. There's no dynamics. There's no rhythms. There's no oscillations. There's no magic.
Yeah, but there are more and more people, for instance, looking at spiking neural networks. That have the spiking neural networks. They have these temporal dynamics. Well, okay, they are spiking.
Yeah. No, that's why in neuroscience and theoretical neuroscience, we do care about all the dynamics, of course. I think in the machine learning, artificial intelligence, at least, I'm not an expert, there's lots of stuff going on. I mean, people do everything, right? But the main trends, like deep learning or whatever, really is, it's more It's not focused on dynamics. It's more focused on, an input-output function that's going to classify or cluster your data. And that's, great. Lots of people are working on that. I think this is more fun because you get, but it's also just a different motivation, right? So here we're motivated by the fact that networks in the brain do more than just cluster, right? We do more than just look at images and decide is that a cat or a dog, right? They have to generate rhythms, oscillations. They have to generate patterns of locomotion. You have to be able to remember songs. There's it's not just kind of static patterns, right? Of course, the machine learning tasks also we'll get there, and different groups are do think about that already. But yeah, we're much more motivated by the sort of networks in the brain. And if you look at networks in the brain, it's like they're constantly oscillating, they're constantly generating all these rich patterns of activity. And so even simple, if you're going to model that, even with simple models like the ones that I present, you want to get at that richness of the dynamics. So.
Yeah, thanks for the wonderful talk. It was very, very interesting. Before I ask my question, let me just comment on what you said on learning dynamics and machine learning and deep learning for dynamical systems. So it is true that there is a large bulk of work on, I'm too close to the speaker, I think, on classification, but you can also think about, trying to predict the next time step given your current step. And there's a very large bulk of literature on reservoir computing and recurrent neural networks, also from a neuroscience point of view, that also try to understand the structure of these networks and different types of fixed points and chaos you can get. So there is actually a very large bulk of work on this. So
and that stuff is close in spirit to what I'm doing.
Absolutely.
But that's coming from theoretical neuroscience community.
Yes. Okay, cool. So I was just wondering, because you showed this is really cool that you can get all of these different limit cycles, but in the actual behaving animal, it can spontaneously transition between these different behaviors. And not only that, there's a lot of variability within the same behavior. So it has actually been shown that in some cases, the dynamics is closer to chaos than it is to a limit cycle. The degree of fluctuations that happen within each of these so-called limit cycles is quite large. So I was wondering whether you could, comment on if it's possible to generate, this sort of variability and spontaneous transitions from a kind of intrinsic point of view in the construction of the network.
Yeah. So sorry. The examples I've been showing are, we're sort of very focused on, we want to model these particular patterns, right, and have them coexist and so on a network. Now, if you take, I didn't include the slide on the talk, but if you take, large networks of these, right, just make a very large graph, but still create the, a CTLN with the same kind of rule, you do see what appear to be spontaneous transitions and sort of large-scale chaotic dynamics. We don't understand it very well, like we don't have nice relationships between, these unstable fixed points are creating this, chaotic attractor or whatever. But just from playing with these networks, that does seem to be something that emerges very naturally. So yeah, unfortunately, I did not include those, but we have, many examples. But it is just at the level of examples now. So we don't have a good theory about it, but we do think that these networks are also supporting that. And in fact, when Hanlazer and Sung and company were studying them early on, they also looked at those questions and viewed them as quite capable of producing the sort of edge of chaos kind of dynamics.
So in the pipeline, there is a question from Bruno Cessac, who can unmute himself from online, and then we have two questions from the audience here.
Hello, can you hear me?
Yes.
Yes, thank you. I have a question. Your hyperplanes somewhat defined a partition of the fast space, and you could be tempted to use that to do symbolic coding, as it's done in ergodic theory and in classical dynamical system, hyperbolic dynamical system. The problem you have is your singularity set, where the dynamics is non-differentiable. I was asking myself, first, if you can write conditions so that the attractor does not sit on the singularity set, so you are away from it. And then I think you can find conditions where on the Wij, where your system is either contracting or expanding, so it's uniformly hyperbolic. And I was thinking if you could use results from ergodic theory to characterize your dynamics. I was especially thinking of the shadowing lemma, which allows you to characterize periodic orbits and recurrent properties that could be helpful in your case.
Yeah, so unfortunately I didn't hear all the details of the question,
sorry.
but I think sorry, it's the connection is not very good, and I'm in sort of an awkward position here. But I think one of the things you said is ergodic theory from classical dynamical systems theory, if that could be used to characterize the dynamics in some of these cases. Yeah. So I'm actually very fortunate to be a professor at Penn State, which is a university that our math department is actually a leader in ergodic theory and pure dynamical systems more generally. And so one of my graduate students, Josh Pike, is actually currently completely pursuing that direction. So he is like learning all the dynamical systems he can from people doing pure ergodic theory and trying to connect it. So I guess the short answer is yes, we think that that is possible, and we're working on it. So,
but my question was, how do you deal with the non-differentiability of your dynamical system?
Yeah, that's the tricky part. Yeah. That's why we can't. I mean, so we talk to these people. I've talked to my colleagues before about this, and it's the system is not set up that you can just use a lot of the results out of the box, right? Yeah. Because it's just not quite right. You don't. There's some hypothesis always in a theorem that you don't satisfy, right? But there's something. Still, it should be adaptable, but it sort of requires more work, and so. I had to kind of wait until I had a graduate student who was really willing to do that. And so, yeah, check back in a couple of years, hopefully.
Thank you.
Yeah,
Thank you.
by the time he defends, we'll have something.
Hey, thanks a lot for this presentation. I come more from neuroscience, so I have more related question because we are in biology used to very complex network, and here you show how you can, in a constructive way, have this multi-stability system. But can you reverse in some way the process? If I give you a very complex network, can you separate some underlying networks that are responsible for a given attractor or stable state?
Can we learn this from data for a complex network? That's the question. So so I think that is also kind of, I hate to say it, but that is also in the future work category for us. So we've been trying, because these networks are tractable enough, right? We actually can address the sort of forward problem. Right? You start with a network, you know everything about it, what is it going to do? Right? And that is hard as well. I mean, so people train networks, even if you go to machine learning, deep learning, people train the network, and then there's this question, okay, suppose someone handed you one of these trained networks, right, and didn't tell you ahead of time what they trained it to do. Could you look at the network and predict what's happening? It's hard, right? And so this, for us, these networks are an opportunity to really tackle the forward problem first, right? Which is what we've been doing for many years now. And the hope is that if you really understand the forward problem better, then you can do the backward problem, right? And so and in some sense, this engineering networks to do specific tasks is trying to do the backwards problem in sort of a simpler setting, right? Where we have, again, sort of control, like, okay, these are the different limit cycles we want, let's engineer a network that does that. But that is sort of a reverse engineering question already. And then, so the longer term goal is that you can look at a data set, right? And apply, and develop some kind of learning rule where you would automatically train the network to reproduce those dynamics. That we do not know how to do yet.
Yeah, thank you very much for the talk. So it was very interesting to separate between initial conditions and structure to go from one fixed point to the other. I have a quick question, maybe it's not fun for you. Did you check for changing the weight to go from one fixed point to the other? So to have a dynamic kind of synaptic plasticity, but just having a rule to change the weight and...
Right, so I mean, and so, oh, sorry, these are other slides that I didn't, that I cut from my talk. So in this example, right, we added noise to the W matrix. So that just meant we added sort of random numbers, we just added a random matrix to the W matrix, whose amplitude, where the amplitude of the individual entries was about 1%, and then we did a 5% and so on. So this is like randomly changing weights, just a little bit, right? We also have a completely different body, related, but that I haven't talked about at all today, body of work where we are trying to understand for a given attractor what weights can be freely changed while maintaining the attractor to be exactly the same. Okay, so this is another kind of cool thing about these networks that and because this is a short talk today, I didn't get to talk about, but we actually can understand using our theory with the unstable fixed points and so on. We're able to, at least in small cases, we're able to say, okay, here is a small network. It has this like motif in it that's responsible for some attractor. Here are some weights where if I alter them, I'm going to screw up the attractor or change it or lose it. And here are other edges that I can freely move and preserve the attractor. And so, so part of it is understanding, like, in the space of W matrices, in the space of TLNs, we are mapping out actually directions that you can move and preserve an attractor. Even
and going from one attractor to the other?
And directions to go from one attractor to another, yeah. Okay. And so, yeah, so there's a whole like interesting bifurcation theory there. I don't know if that answers your question, but this is definitely something, yeah, that we're looking at. I just didn't get a chance to mention it. Mm-hmm.
Thank you, Karina, for this very nice and very dense talk. There was a lot of material. I have two questions. One is about cascades, and one is about bifurcations. So cascading, the idea of having this, multi-layers networks. Can you actually, in some cases or every time, predict the dynamical behavior of the cascade of networks from your theory? I was under the impression that in some cases the answer was yes.
In some cases, yeah. It depends on what you. You're thinking of a network that has some kind of feed-forward structure, or yeah. So in fact, so there are different ways of defining the feed-forward structure. So we actually have this sort of like dynamic feed-forward structure that we define, and that's the last part of my talk I didn't get to. We're thinking about these directional graphs, and what makes a graph directional is that there's a subset of the nodes that supports all of the fixed points. Okay, so you have a large network, but then all the fixed points are supported on some subset. Okay, stable and unstable, right? And what we observe, and this is heuristic, is that the activity tends to collapse onto that subset. And then you string many of these together, you get like a cascading, and this is what we have with the directional graph. So this is sort of a cartoon example of that. And yes, we can say a lot in those cases.
I had a quickie. I was under the impression that your family of weights was a two-parameter family. You had epsilon and delta.
Yeah.
Have you thought of looking for bifurcations of your system of equations? 2 parameters is very small.
Yeah. Yes. Yes. We have. In fact, 2 parameters are very small, and so
it's non-differentiable, of course. It's piecewise linear.
It's piecewise linear, but. Yes, so the piecewise linear, so it's non-differentiable, so that's annoying for certain tools, right? But on the other hand, then you still have this local linearity, right? Which is very nice and very convenient for other, for other tools. So yes, we do have, in fact, I cut all these slides out, but no, sorry, it's not here. But yes, for instance, the limit cycle that emerges from a cycle in the graph, that one can be understood with a hop bifurcation, even though the hop bifurcation theorem doesn't apply. The limit, no, it doesn't apply because locally your system is linear, so you don't predict that you get a limit cycle when your fixed point goes from stable to unstable. But that's when the limit cycle emerges. Even though the limit cycle is cutting through different hyperplanes and so on, it's emerging at the same time, right? So again, it's with the bifurcation theory, like many of the theorems, of course, we can't just apply straight out of the box, but there's a spirit of them that we observe is very similar, and so there should be some way of adapting. Yeah. Yeah.
First off, let us thank Professor Courteau and Dr. de Guerriere for the very nice talk of this afternoon. The session will reprise in 30 minutes, right? 4:30. Second part of the Algebra and Geometry afternoon. It is my great pleasure to introduce Lida Canari and Catherine Hess from EPFL Lausanne. They're going to talk about a topological insight on neuronal morphologies. Lida.
Thank you for the introduction, and I would like to thank also the organizer for giving us this opportunity to participate to this very interesting conference. Unfortunately, we were planning to be in Paris, but that didn't work out, so we'll have to do it to present from Lausanne. But we've heard today in this session two talks that were in very, very different levels. The one was in microscopic level and the other was in network level. I think our talk is somewhere in the middle, so we're trying to link those two levels, and I'll discuss how in a little bit. So we're going to talk about the topological insights on neuronal morphologies. And in order to do that, I will first describe what are the neuronal morphologies, how they're important for the rest of the networks, and how they work within the brain, and then I will give some information about our work with Catherine on the topic of neuronal morphologies. So here is a picture that we're kind of trying to put together all the pieces that we have information about in the brain. And we start from very, very small level, such as ion channels and the vesicles that we've seen topic earlier today, which are populating neuronal morphologies. And these neuronal morphologies create dynamical properties, such as the electrophysiological traces that we can see. And together they form brain regions that create the dynamical properties of larger networks, and eventually they give rise to the network activity that we observe in the brain. But let's start a little bit from the broader picture, and then we'll dive in. So first of all, what are the anatomical properties that are important for a brain? You see here two different pictures. On the left side, you see a picture, an actual picture from the microscope of a brain slice, where you see the different brain regions. And what we're trying to do in the Blue Brain project is we're trying to reproduce as many details as possible computationally of these anatomical properties that we observe in the microscope and in the labs. So this is the left picture was taken in the lab, where you see the different regions, and if you zoom in really into the details, you see the somata positions, so where the neurons are located in these different regions. And on the right side, you see a model developed by a PhD student in the Blue Brain, in which you recreate these positions within the brain. So once you have these positions, what you have is a density of neurons and other brain cells within the brain. But these cells are not really like points. If we delve into each one of these positions, they have these very distinct and complicated branching structures. And each one of these morphologies has a unique structure. However, what we can do to try to understand better this very, very complicated system is to categorize these morphologies into distinct categories. So we are trying to link properties that are shared among many morphologies to define some groups of morphological cell types that will give rise to their functional role within the brain. And starting for a very broad distinction, we have inhibitory and excitatory cells. So you've seen in the previous talk how inhibition and excitation can contribute to a large network. But here we're just trying to see what the different shapes of these two neuronal types are contributing into the whole picture. Once you have the categorization of the structural types, so the morphological cell types, then you need to go into the understanding of the dynamical properties of each one of these single neurons. And the dynamical properties is, if you simulate one of the neurons, you see different electrical responses. And here is just a small subset of example traces that you can get from all these different types of neurons. And just to clarify, we are in our lab, in the Blue Brain project, we are only studying a very, very specific subset of the brain. So we currently, the work we have done is focused on the cortical neurons. And as you see on the right side on the C panel, these are the combination of different morphological and electrical combination of types that we can have into the cortex of the rodent brain. So you can imagine how broad this will be if we were trying to study all these types for the whole brain. So the question is, why are neuronal shapes so important? And to understand that, we need to understand that these shapes give rise to the electrical behavior of the neurons. So they define very specifically what the electrical responses will be from different properties of their shapes. A very simple example for that is you've seen all these different shapes in terms of branching, but what is not clear is that the thickness of these branches is having a huge contribution on how the neurons will behave electrically. So even the components that are not clear just by looking at the larger picture are playing a very significant role on how each of these neurons responds. In terms of connectivity, so in the larger picture, the neuronal morphologies constrain the connectivity because they define the connection points between axons and dendrites. So in order to have all these very complicated network dynamics that we see in the brain, you need to have these specific neuronal shapes that give rise to these different network dynamics. And this is pretty much the way that these two components, how neuronal morphologies shape the dynamical properties of brain networks on a large scale. And here is a very nice sketch, which is probably one of the first representation of neural morphologies by Ramon y Cajal. And that's how he started studying, looking at the morphologies and drawing them by hand. And now we are more at the stage where we can have digital versions of these morphologies. Here on the left, you see examples of different shapes of morphologies within the brain. And as I mentioned before, you can have very, very different branching patterns that we see. So this is somehow similar to the botanical trees that we see in nature. And as we have a very large diversity of trees in nature, we have very large diversity of neural morphologies in the brain. So when I started my work in the Blue Brain, I was tasked with identifying different types of morphologies. And I was specifically given these two and many more layer 5 pyramidal cells. So those are excitatory cells. And the experts were telling me that they are clearly different. But when I was asked, like, how can you prove this mathematically that there is a difference? To my eyes, there was no clear difference. So it was very hard to understand what was different from my perspective, even though for the experts that were in the lab tracing these neurons, this was evidence. So what we did with, in collaboration with Catherine, and inspired with the topological data analysis methods, was to find a way to encode the information that you see into this branching pattern into a topological description. And the motivation for that is that in the past, neuroanatomists were trying to measure specific properties. So they were measuring, for example, the length of the branches, the angles of the branches, the number of the branches, but all these are one-dimensional projections. So you were losing so much information from each one of these projections that you couldn't capture the full picture of the neuron. So what we were trying to do is to find a descriptor that would capture as many information as possible from these neurons. And the idea was, is coming from persistence homology. And the idea was to trace back each one of these branches and see how they connect to the root. And if we take as an example, so you can do that with many different types of distances, but if we can take as an example the radial distance. So here you see a very simple tree. And if you take any two of these tips of the tree and you follow it back towards the root, at each bifurcation point, you check between the two points. So for example, 2 and 3, which one has the largest radial distance? And 2 is further away from the soma, so 2 persists and 3 creates one bar here in the persistence barcode. So this way we can follow back all the terminal points of the tree until we reach the root of which is the soma of the neuron. And this decomposes the neuron into these bars within the barcode, which represent the whole tree. An equivalent way to look at that is if we take the start and the end point of each one of these components within the tree, and we represent it in 2D. And here is like the start point is on one axis and the termination point is on the other axis. So each one of these points corresponds to one of the bars that we've seen before. And this is useful because then it allows us to average different trees, and by allowing Gaussian kernels around each one of the points within this persistence diagram. So this is pretty much setting up the background of what I'm going to use as tools to analyze neuronal morphologies. And here is an actual neuronal morphology, and you see this process of following back each one of these tips and decomposing this neuronal tree into the barcode. And as you see, we follow each one of the little smaller tips, and then we have the largest component, which generates the largest bar in the barcode. And as I mentioned before, we can take each one of these points and represent it into the persistence barcode. And then at the end, we can generate the persistence image by allowing Gaussian kernels around each point. So this is creating a summary that is capturing the branching properties of the neuronal tree. And we're going to use that for many different applications. Now, if you'd allow me to go a little bit into the mathematics, so this is, we call it the topological neural morphology, so I will refer to it as the TMD from now on. And we wanted to prove that the TMD is stable with respect to some perturbations. And these perturbations are motivated by the errors that we see in the experimental procedures. And one of them is the error in measuring the exact position of the nodes. So when you click and you make these reconstructions, you don't know that you got exactly the correct position. And the second one is you can have smaller branches that you don't see, or errors in the image that create branches that are not there. But those are the two types of perturbations that we wanted our algorithm to be stable against. And so we proved that the bottleneck distance between two pairs of trees that are with respect to these type of perturbations that I described before is bounded by epsilon, so by the size of these perturbations. So this was a very good example, a very good result, because then we can use this algorithm on the experimental data, even though they have intrinsically some of these errors in them. but the difference in the TMD that they're producing will not be significantly large, so we can use them without any like big effect on the results that we get. So if we go back to the previous example and we perform all the actions that I described before, so we get the barcodes, and from these we represent it as persistence diagrams, and now we get the persistence images of the two trees. Now I think the difference is much more striking. So the experts were calling these type of trees tufted, because as you can see in the persistence diagram, in the persistence image, sorry, there is a tuft that is now very clear, and it represents that part of the tree. And the other type of neurons were the untufted, because this tuft is missing. So this is something we can now see very clearly with this decomposition into the topological descriptor that I described. But what is even more important is now we can actually do statistics on these type of descriptors. And one very, very simple operation is to illustrate what are the differences between these two trees. So in order to do that, we can subtract one persistence image from the other. And as you can see here, we get a higher density of branches at this location, which indicate the tuft that we've seen by eye. But interestingly, we see a difference also on the branches that are around the soma, which is not something that you can detect just by looking at the two pictures. So we have identified, for example, that here, the first effect is the existence or absence of a tuft, but also the branches around the soma have a different topology. So they're longer in this tree compared to this tree. And this is actually going to be interesting for a few examples that I will illustrate afterwards. So I will describe a few of the applications of these methods. So how can we use the TMD to classify cells, for example, to cluster them, and to finally for the inverse problem. And I'll start by the classification of rat pyramidal cells. So these are examples of excitatory cells within the rodent cortex, specifically rat. And the task was to find an algorithm to objectively separate these cell types, but also to try to explain what is different between these cell types. And here is an example of the layer 5 pyramidal cells that I described before. So here are just unique morphologies from these populations, but you see the persistent images of many, many cells, like 50 of the same cells. And as you can see, we have a very distinct pattern in which we have a tuft in some cases, in these two specifically. So these are large tufted pyramidal cells. Then we have the tuft that has a different shape, which is much smaller. And these are termed as small pyramidal cells. And then finally, we have pyramidal cells that are lacking completely this tuft, and they are untufted cells. So this way we could objectively classify the neuronal types within the rat. And we can also, this is a way to also understand what is different between the classes that we see. Here is an example. So in the cortex, we have all these cortical layers from layer 1, which is the superficial one, up to layer 6, which is the deepest one. And these are the classes that we have identified, and the names were given by experts. But as you can see, we get a very good classification accuracy for the main classes. Here is a more mathematically interesting example of we can use, so you see the different layers. So we're trying to classify the cell types, the excitatory cell types within each cortical layer. But what is interesting is depending on the distance that we use mathematically, we can have a very good classification, or we can have more vague results. And in general, the persistence emojis that you've seen before, so here, these type of persistence emojis were very, very good at distinguishing all the layers, and their differences could be identified, and like we could pinpoint the differences between the different cell types based on the Gaussian emojis. As you can see, there are other type of distances such as the landscape, based on the landscapes, that are also pretty good at distinguishing cell types. But the bottleneck, which is one-dimensional like projection, is losing information, so it's not able to distinguish cell types, so the accuracy is much lower in this case. Now, another interesting example is clustering, and the difference between classification and clustering is that in the classification task, we have the classes. So we want to see if we can objectively justify them and identify their differences. But in clustering, you have no classes. So it's like unsupervised learning. And the task here is, we had some very nice neurons from human cortex. And we wanted to find if there is any distinct shapes within these neurons. And again, just by looking at them, it's very hard to justify that there would be 2 or 5 classes or whatever number of classes. And in fact, the experts here were not able to agree on which are the classes in which these neurons should be separated in. But if you do the persistence emojis again, you see a very, very striking difference. So you see that we have 2 populations that are very distinct in the human cells. Again, it's related with the shape and the existence or absence of these tufts. And as you can see, some of them are very slim. So we call these slim tufted cells, and the other ones have a very nice and profuse tuft, which we call profuse tufting. And what we wanted to identify is if it's a clear separation. So do we have these 2 classes, or do we have also intermediate classes in between? So I trained the classifier based on these examples, and then I extracted the probability that each one of these neurons that you've seen before, I think there are around 100, the probability that they fall within these 2 classes. And only 5% was not able to fall into these 2 classes. So that means there is a transition between these 2 classes, but it's very slim. So you see a very clear separation within this data set in these 2 classes. And what is interesting here, once we did this analysis, we were lucky enough to have the electrical traces of the same data set. So the electrical traces of each neuron that we've seen before. But once we did this separation based purely on their shapes, we saw that they exhibit unique electrical properties. So they fire at different rates, which is something we are still trying to figure out why this is happening and how these 2 types are different. But apparently the shape is important for their firing properties. So we use topology to group neurons so far. And now I want to show you a new example of morphological phenotypes of microglia. So apart from the neurons, we have other cell types in the brain. And one of them is the microglia, which are related to disease and lesions in the brain, and the metabolic paths that you have. So here you see an example of different brain regions, and these are taken like photos taken from the microscope, in which the group of Sandra Sigurd that we have a collaboration with in IST, they took these images from the brain regions of the different brain regions, and they identified that the shapes of microglia in these regions are very, very different. And they could also reconstruct single microglia from these regions. But then the idea was to analyze them using persistence homology. So once again, we extracted the persistence images for each one of these regions. But here we have an important problem. So microglia are very dynamic because they respond to the metabolic pathways within the brain. They change a lot within very small time frames. And the result of that is that we don't have a very specific shape. We have representative shapes and a high variation within these shapes. So the idea was to try to bootstrap in order the pool of microglia in order to create more robust signatures that would allow us to recognize differences between each brain region. And once we did this process, you can see here that the separation of the different shapes of the microglia for these different regions is it's very distinct. And this was something that was expected from this image that you see because they look very, very different. So if you reconstruct each one of these microglia, you would expect to see a significant difference in their topology. However, this was not clear when you just study the shapes of single morphologies. As you can see here, they don't look that different. But what is even more interesting as a result is in this amazing data set that we were given from Sandra Sigurd and her group, there was a difference between, they were annotating the sex of the mice. So there was a difference between male and female. And if you do the same decomposition that we did for brain regions for one set of animals, so we put them together, but now we separate males and females, we see that the representation of each brain region in this hyperspace is very different. So what this tells us is that there is a different distinct pattern in the microglia within the brain, depending on the region, that can separate male from female microglia, which is interesting because I'm not showing that here, but they went deeper into this analysis and they studied how the microglia respond to different lesions. And what you could see there is that the male and female microglia respond differently and recover differently for different conditions. So that could explain why some brain diseases are expressed differently in males and females that are related to the microglia shapes. And I think one of the main reasons to study microglia is because of Alzheimer's disease. They're highly related to Alzheimer's disease. So it would be very interesting to see what this connection would be with microglia shapes. And this brings me to the final application that I will present, which is the inverse problem. So what we did so far is to create from neuronal morphologies topological shapes and to use them in order to understand better the neurons from which they come. Now, what we want to do is, if we have these topological shapes, can we create neurons? Can we recreate the shapes from which they came from? And initially, that was a question of encoding. So if you have a specific barcode, can you get the exact neuronal tree from which it came from? But what we noticed is that this is not the case. So we cannot get a single tree that will, gives rise to barcodes. So it's not unique to go from the trees to the barcodes and back. And Catherine will talk more about this relation later on. But what I'm going to showcase here is we are using this inability to go back to single tree to create a population of different trees that are, each one of these trees is unique, but they have the same topological properties. So how can we do that? We start from the soma, and we start growing some like a random walk. So some type of directed random walk, and at each point, we have a probability to continue, to bifurcate, or to terminate. And if we couple these probabilities to bifurcate and to terminate to the barcodes, we can recreate shapes that have trees that have the same topology. So what we can do, we start from the root, and as the branch grows, a branch has a probability to bifurcate at each distance, which depends exponentially on the start points of these bars. So as you approach the distance from the soma for each one of these bars, your probability to bifurcate increases. And then it has very similarly a probability to terminate. So these branches, as you grow, are gonna have a probability that to terminate that increases as you get to the end of these bars. So imagine if you only had one bar, it would start growing point by point, and it will just terminate at the end of the bar. So you will only have one neuronal branch that you could generate. But if you have multiple of them, this probability accumulates, so you can generate branching shapes that have very similar barcodes. And here is an example from the analysis that we did before, pyramidal cells of the rodent cortex. So here you see all the different types that we have of pyramidal cells that we have identified objectively. And in blue, you see the original cells. So these are random examples of the representative cell types. And in red, you see the synthesized, so the computationally generated. So use this method, and depending on the branching properties of each cell type, we could generate very, very different shapes. The important point here is that we didn't need to fine-tune the algorithm based on the different shapes that we see. So here, for example, you have these large trees with the very intense branching far away from the soma. But both of these and this one, which is super simple and it doesn't have any branch far away from the soma, both of them could be generated with exactly the same algorithm. The difference is captured in the topology that is it's sufficient to just have the barcodes of these different types in order to generate the different shapes that we observe in biology. And one of the reasons we want to generate these different shapes is because we want to create models of computational models of whole brain regions that are accurate up to the details of morphologies at least. So what you see here on the right is an example of a cortical column in which we put together the artificially generated neurons, and the different colors are the layers the cortical layers that I described before. And as you can see, we get a soup of all these morphologies that is, and I'm going to show you later how we validate that, but it's very similar to the observed microcircuits that we have in biology. So we need to make sure that the neurons that we're computationally generating work correctly. So the first step, apart from the shapes that we just see by eye, is to electrically simulate them. And the idea here is that we have tools to go into the soma, induce some current, and see what are the electrical traces that they produce. And we performed this experiment for a number of different reconstructed and synthesized morphologies, and we see that they produce very, very similar traces. And this was just a single example, but we performed that into all the different morphological and electrical types that we have, and we also performed a statistical analysis for these in order to verify that all the different features of these electrical traces are correctly reproduced. Similar to that, we want to study the morphological properties of the cells. So we want to verify that if we generate from 50 cells of a specific type, so these are specifically layer 5 pyramidal cells. So if we generate from these 50 cells any number of artificially generated cells, if they will have the same properties. And here you see just a small subset of these properties in which the synthesized and the reconstructed cells have very similar behavior. So this is in order to make sure that the shapes that we generate artificially follow the properties of single neurons within the brain. However, that's not sufficient, so that's not really what we are aiming at doing. What we want to do is to be able to manipulate the barcodes in a way that we can generate shapes that we are not able to get examples from. And a very, very easy illustration of that is within the cortex, we have different cortical thickness. So we have different thickness as we move along the cortex. And this thickness of the cortex defines the size of the cells that live within this. So a very, very easy way to manipulate the cells is just to change their barcodes in a consistent way with respect to the cortical thickness. So as you can see here, we can start from a region in the cortex that's similar to where the cells were sampled from. And as we move along the cortex to similar sizes, we make the barcodes smaller and smaller, and we can generate cells of all the different types that have similar branching properties, but very different sizes. So we preserve the branching, but we change one component here, that is the size of the cells. So this might look very simplistic, but it's a starting point for something very, very for an interesting application that we're very excited about. So here is, again, a very, very simple modification that we're going to perform on the dendrites. So here is an example of an actual dendrite and the connectivity of the network that is formed in a column like this one that you see. So this is the connectivity that we get from a cortical column, and this is the connectivity between all the cell types that compose this column. Now, one of the modifications we can do is to take these structures and cut branches and generate a column based on similar cell types, but with smaller number of branches. And interestingly, what we see here is if we strip, so this is pretty simple to implement, because in the barcodes, we just keep the longest bars, and we start reducing the number of bars that we're using in order to generate synthesized cells. So interestingly, if we do that to all the cell types, and we end up with very simple dendrites, but the connectivity between the different cell types preserves pretty nicely, which means that even if you lose a lot of branches, the cells are able to connect to the different regions because the main branch is still reaching the cortical layers that it's supposed to. However, let's perform another very simple, again, operation, and that is shrink the dendrites. We take, again, all the cell types and we shrink them all at the same level. What you see here is even at, if I remember correctly, at 30% shrinkage, we were losing so many connections that we couldn't reproduce a network. So this is an indication that the way that you change the branching structure is going to have a huge effect on the network that these neurons form. And of course, as I said in the beginning, these are very, very simplistic operations on the dendrites, but our goal here is to try to reproduce specific dendritic lesions that we see in the brain, and they are associated to specific brain disorders, such as stress disorders. So the shrinking of the branches was motivated because in many stress disorders, what they observe in biology is that the branches retract quite a lot close to the soma. So this could potentially explain how the networks collapse if the changes of the, based on the changes of single dendrites. And here is a statistical analysis of the phenomenon that I described before. So you see that in case where the branches are cut, so you end up with having just the backbone of the neurons, the connectivity collapses much more slowly. In case where the dendrites are shrunk, what happens is that the connectivity collapses immediately. So you see a huge effect on the networks if you're shrinking the dendrites. And to summarize this part, so this is the idea to use topology to synthesize the router cortex. So we go from single trees to the topological barcodes and of each cell type, and we are recreating from this the cortical column that you've seen before. And even more impressively, now we can have the first brain region, which is completely generated artificially. And this is the rodent neocortex. So I'll pass on to Catherine, who's going to talk more about the mathematics behind what you've seen so far.
Thank you, Lila. So yes, I'm going to take over now since I'm the mathematician, or the purely mathematician in this story. So I want to talk about how we can use these sorts of tools to learn something about how we can distinguish neuronal trees from other trees, because you could think about a neuronal tree as being some sort of combinatorial tree embedded in 3 space. And then you can say, well, is there something particular about the sorts of geometric trees like this that you get from when you're looking at apical dendrites of neurons? And if you want to think about, for example, different species, or you're thinking about different brain regions, are you going to see differences in the kinds of populations of neural trees? And can that be captured by this sort of topological signature that we're looking at when we consider barcodes? So you would consider, as is illustrated schematically here, your cortical column is being populated by barcodes rather than by neurons. And we want to see, is there something, so instead of studying the hard problem of saying what's so special about the tree structures that are showing up in neurons, you can make it more simple and more purely mathematical and say, well, is there something special about the barcodes that arise from neuronal trees of this sort? So next slide, please, Lita. Right, so if you think about a barcode, so here we have a figure illustrating what we call the permutation equivalence classes of all barcodes with 4 bars here. So when you think about these permutation equivalence classes, what we mean is if you look at one of these barcodes, so we've arranged all of these barcodes so that the birth times of the bars, the beginning point of each bar, they're ordered increasingly. And then you can look at the other side to see what is the order of the endpoints of the bars. And this gives you a way of associating a permutation to each barcode. And it turns out that just looking simply at the permutation associated to a barcode, it actually contains a lot of information about your particular barcode. And it's interesting to classify the barcodes by their associated permutations. And we call these the permutation equivalence classes. And so one very simple representation of something as complex as a geometric tree, or the tree associated to a neuron, or a dendrite in any case, is just the permutation equivalence class. So what permutation do you associate to its barcode? So next slide, please, Lida. And this gives us a way to think about how we can answer the question of whether this topological neuron synthesis that Lida was talking about, where you go from a barcode and in a stochastic manner, you're taking into account an exponential probability of bifurcating or terminating, whether this topological neuron synthesis gives you some way of having a kind of an inverse to the TMD. So what's illustrated in this figure here is on the top, the idea is you start with a barcode, and via this topological neuron synthesis, you're going to create some sort of a geometric tree. And then the question is, of course, there's a stochastic element here, so you're not always going to get the same tree. You're going to get, there's a sort of a distribution of possible trees that you can get. And then you can say, okay, I go from the barcode to the tree, and then I can again apply the topological morphology descriptor and get a new barcode. And I can say, how different is the new barcode I obtained from the one that I started with? On the other hand, you could start with a tree, a neuron, a dendrite, look at its associated barcode, and then apply the topological neuron synthesis and get a new tree. And you can say, okay, how different is the tree that I obtained in this manner from the one that I started with? And so what we did in work with Adélie Garin, who is one of my PhD students, is to apply techniques from, well, not only topology, but also a bit of statistics and probability to be able to answer these questions. To what extent topological neural synthesis is some kind of inverse to the topological morphology descriptor? So the first question is, we can formulate then in terms of what we call the stability of topological neuron synthesis with respect to bottleneck distance. So here, this is the idea of saying we start with a barcode, we look at an associated tree, and then we get a new barcode from that. So we start with the barcode, apply the topological neuron synthesis, it gives us a new neuron, and then we look at the associated barcode. And the question is, how different are the two barcodes, the one you started with and then the one that you get from output? And the notion of distance that one uses between barcodes, so there are a number of possible distances. One of the most commonly used distances is something called the bottleneck distance, which is a sort of optimal transport distance. And so we said, okay, let's look at how far apart these are in terms of the bottleneck distance. Now, if you remember, perhaps from the description of the topological neuron synthesis, there's an important parameter in this construction, which is called lambda, which has to do with, which parameterizes the probability of bifurcation or termination, depending on how far you are from one of the endpoints of the bars in the barcode for which you're constructing an associated tree. And the value of lambda that produces geometric trees that look the most like actual neurons is lambda is 1. So this is in units that have to do with the step size of the random walk that you're making as you create this neuron. And so if we focus here on the case when lambda is 1, then we're looking in this plot at the probability that the bottleneck distance between the two barcodes, the one you started with and the one you created by doing topological neuron synthesis and coming back down, is greater than epsilon. So when epsilon is very large, in the case when lambda is 1, which is the blue curve here, we see that the probability that the bottleneck distance is larger than epsilon is actually very small until you start getting to small distance to epsilon. So it's a sort of stability which says that the barcodes that you're going to get, if you go through this process, the new one will, in general, not be too different from the one that you started with. So this is some way of expressing stability of this topological neuron synthesis. Next one, please. And this is a bit different, whereas another kind of stability that we were considering in the case where I said if you look at these barcodes where you have ordered the starting points so that they're increasing order like this, and then we had some sort of permutation coming from looking at the order of the endpoints with respect to the order of the starting points. Well, you could change that order by sort of switching the lengths of two of the bars. And what you want to see is also, if you're looking at this process of starting with a barcode, looking at the associated tree, by the topological neuron synthesis, and then calculating the topological morphology descriptor, so getting a new barcode out of it, you could say, what is the probability that you're actually going to have two bars that change order like that? So again, focusing on the lambda equals 1 case, which is the most relevant case for us, we see that the probability that two bars will swap places like this when you go through this process of using topological neuron synthesis and then looking at the associated barcode, the probability that you will have two bars that have swapped positions, swapped orders in this process is actually, it's a function of the bars. So the greater the distance between the endpoints of the bars, the lower the probability that they will switch through this process, which is what you would expect. And so this is confirming this nicely. And particularly in the case that we're interested in, which is this biologically relevant parameter lambda is 1, then this the blue curve here. And again, we see that the probability of having a change, a swap in order like that is quite small, as long as the distance between the bars is also very small, between the endpoints of the bars, more like. So as I said, it's sort of, you can go ahead later. But one of the motivating reasons we wanted to think about this was to say, well, what can we say, what is special about the barcodes that are associated to actual neuronal trees? So what we did was, for a very large number of reconstructed neurons, looked at both their basal dendrites, so the sort of root system of dendrites that's around the base of the, gathered on the soma, and the apical dendrites, which are the ones that usually reach towards the surface of the brain, and looked at those as different neuronal trees and said, okay, well, let's look at their associated barcodes. And then we have something called the realization number of a barcode, which is how many combinatorial types of trees can have that particular barcode. So that's called the tree realization number of a barcode. And then we plotted based on how many branches or how many there were in the tree, or equivalently how many bars there are in the barcode, the realization number of these. And so in each of these, we're comparing, looking at the basal dendrites and apical dendrites. The red curve is saying what is the maximum logarithm of the realization number. So the largest, as a function of the number of branches or the number of bars, it's always n factorial. That's why we take the log here, because it goes very fast. But then to say, okay, but if we look at barcodes that are chosen at random, what sort of realization number are we going to get? And that's what the yellowish, yellow-green points are here. So, and plotting that number of bars against their realization numbers. And then the blue points are coming from actual biological trees, coming from either the basal dendrites and apical dendrites. The basal dendrites seem to be more or less like random trees, but the apical dendrites are different. They have significantly lower realization numbers. So the number of possible combinatorial types of trees you could have with that particular barcode. So there is something special about the nature of these apical dendrites. And this came out in other ways when we were looking, for example, at the permutations that were associated to these barcodes and so on. So this really begs the question of, again, if we were looking at different species, if we're looking at different brain regions, if we're looking at what happens in disease, is this something that we could characterize or use in a way to distinguish these different states? So that was, I think, the last slide for me, indeed. So I think together with Lida, we would like very much to acknowledge all the wonderful collaborators we've had over the years. Lida and I started working on this together, these projects together back in 2015. At the time, we were working closely with Pavel Lutko, Ran Levy, and Martina Scolamiero, and others. More recently, we've worked with my PhD student, Adélie Guérin. We would like to recognize Henri Markham, who's the head of the Blue Brain Project. And as I was looking at these slides earlier today, I realized we need to put some pictures up there of our collaborators at IST as well, because this most recent project with microglia is very exciting. It's really cool to see the difference between males and females coming out. But in any case, I'd like to take this opportunity to acknowledge the pleasure that I've had over the last 7 years in collaborating with Lida on this really exciting project. Thank you very much.
I hope you can hear the applause for a remarkable talk. Any questions from the in-person audience?
Thank you very much for this interesting talk. I have two questions. The first one It's related to, well, both of them are related to rather to the first part, maybe to Lida. When you obtain those persistence diagrams from dendrite trees, did you ever, I don't know, experience any problems due to the crossings, not bifurcations, but crossings which might appear between, for example, two branches bifurcating from a certain node? They cross each other and they continue still, and they terminate a point which is further from the point where they crossed each other. Maybe on two-dimensional, let's say, figures or images of neurons. They cannot be distinguished, but we can imagine that in the three-dimensional space, in the real case, they might going over each other, and then they might still continue and terminate at a certain point. So I don't know if it was important for the morphological categorization which you made for the neural shapes, but
yes, so I can quickly reply to that because I did not go into the details of the process, but we are lucky enough to have three-dimensional shapes. We don't do it on the images, but we do the process on the reconstructions. So we have the three-dimensional shapes. So that's why we do not encounter these type of problems.
Okay, thank you. And the second question is, where you showed that this topological morphology descriptor idea is robust, stable against small perturbations. You showed quickly this theorem, where you mentioned that a certain kind of distance between two types, between the original one and the perturbed version. Well, a measurement of a global measurement of two versions of, let's say, graph, or graphs of neurons. What kind of distance it is, and can it be applied to no matter what kind of network or graph? Or are there some certain conditions which make the use of such a distance function, such a global measure, available?
So the distance we used is the distance that Catherine mentioned. It's the bottleneck distance, and it's a topological distance. To define the distance in the space of trees was kind of hard. That was the main reason we tried to do it in the topological space. So this is related to the persistence diagrams, this distance, not to the trees directly.
And there are many known ways to define distances between trees and between embedded trees as well. But because the idea was to look in the end at these topological signatures, which are the topological morphology descriptors, it was relevant to use some well-known and well-studied notion of distance between the barcodes, and then to use some relevant, biologically relevant notion of perturbation, of having two geometric trees that are relatively similar to each other. And that's why that was the way we formulated this result. But there are other notions of distance one could use that can be more relevant in other contexts.
Yeah, thank you for the lovely talk. I want to ask, I think you did talk about this, but I wasn't completely clear about how your methods take it into account, which is that it seems like the most important aspect of neuronal morphology, and this is certainly true in the retina, is which layers are connected, from one to the other. And so I wondered how you're taking the layer into account in your initial barcode, and whether you showed very beautifully that that by scaling, right, by the thickness of the cortex, for example, you can get dramatically different changes. So do you always take into account layer structure in the original setup of the coordinate system?
Yes, that's a very important point indeed, that we have information on the cortical depth for each neuron. So we know up to some uncertainty the layer from which the neurons are coming from. So this is indeed a very important parameter in the way that we construct the column and in the way that we analyze the data coming from the lab.
I guess it would be cool, right, if your heat maps of shape sort of pulled out which layer is connected to which layer, you know, where it originates, where it tufts. So a distance along that diagonal, for example, if that told you which layer, because for the retina, you know, what you see with the retina is that it just tufts, you know, straight, right, into two different layers. And so if you clustered retinal neurons that way, retinal ganglion cells, they would just be sort of points in a two-dimensional space according to which layer was connected to which. But in your measure, you don't take that into account directly in the barcode. Is that right? That you can't see any layer structure directly from the barcode.
Yes, so we don't take that into account. If you do it for the excitatory cells, you can separate layers because primarily because of their sizes. But for inhibitory cells, you can't really tell the layer just from the barcode. So this is a component that we add externally. It's not within the barcode.
Any more questions from the audience?
Yeah, sorry. So I have still a question about this choice of the distance, or the bottleneck distance. So I understood that since it is a well-known distance and well-studied distance, it's natural to use it. But so I'm not really familiar with the data, but I was just wondering, since the raw data you have are trees, why not use directly a tree distance rather than bottleneck distance, which is more associated to the topology? But because from my point of view, these trees in our topology, they are used to describe a more complex structure. So it's just a way to reduce the dimension of the problem. But for you, the data are already trees, so why not use a tree distance? Maybe it's a stupid question.
Are you referring to the stability theorem that we were discussing, or in general? Because in general, so there are very different answers to that. In the stability theorem, we wanted to see if you have different trees, how different the barcodes that will be generated. So that was the reason we used a bottleneck distance. In general, in the classification tasks, we are not really using bottleneck because this is a very simple distance, so it does not give enough information, as you correctly pointed out. But we're using distance between the persistence images. We are not using distances directly on the trees because, first of all, they're computationally very, very expensive. So the most well-known distance is the edit distance between trees. And if you try to compute that, it's supposed to be NP hard. So there is no algorithm to do that. And in addition, the edit distance is not taking into account the geometry. It's just taking into account the decomposition of one tree into the other by changing the branching structure. So this was not something that was easy to use directly. And this was actually the reason we made this type of decomposition into the barcodes that are much easier to study, and there are well-known tools to study distances between them.
All right, thanks.
There is at least one more question from the audience.
Hello, thank you for this very nice talk. Maybe I missed it, but I was wondering if the synapses were important in your study. I think because it's important for the connection between the layers. And if you take it into account really in the analysis, and how you would place the synapses on the tree.
So in our analysis, we do not have this information, so we don't get, unfortunately, the locations of the synapses. This is something we have been discussing with a few different experimental groups to try to see, for example, spine locations and try to see what are the differences for spine locations, but for synapses, we don't have this information. Now for the resulting circuits that we generate, this is a completely different team in my lab that is doing that. So this is a different team in the Blue Brain project that they're assigning synapses and then all the synaptic properties. So I'm not at all an expert to talk about that. We do have it in the network models that we generate, but that's not something that I worked on.
There is one more question from the audience.
Thank you. I'd like to know how the molecular classification overlaps with the morphological classification, because in the cortex you have multiple inhibitory subtypes like somatostatin, VIP neurons. Can you tell what is the molecular subtype just looking for the morphology?
So that's a very good question, and that's the direction we are going to at the moment. So we're trying to link transcriptomic information, and there is a very good data set from the Allen Brain project in Seattle, in which they have transcriptomics with morphologies and electrophysiology. So we are going to use that to see how the phenotype correlates with the transcriptomic and molecular information. We have indication that for some big classes that's the case, and we can see, for example, Martinotty cells being very well separated from basket cells, which have different molecular signatures. But I can't reply yet for every molecular information that we can get from this analysis, because we haven't finished these analyses yet.
One last chance to ask questions. If not, let's thank Dr. Canary and Professor Hess for a great talk. So I think this is the end for today, but perhaps you want to say something about tomorrow?
Sorry, it's more a question for Lida and Catherine, because you are online. I was wondering if you would be with us for tomorrow morning, or if you cannot make it. Yes, you could be. Oh, that's fantastic.
We'll be
Thank
here,
you so
be here
much.
Tomorrow morning. Yeah.
Thank you so much. So tomorrow morning, this is as today, 1 hour with the talks of more young people, and then we have this discussion. And as I said this morning, I really encourage all the young to ask questions, because we have this fantastic expert from the Blue Brain project, and I'm sure you will be happy to interact with them. Okay, so thank you so much for this wonderful talk. Bye.
Thank you. Thank you so much for inviting us.
Thank you.
Have a nice evening.
Bye.
Bye. Thank you.